what's the difference production. Today we're looking at types of immunity, and of course I am Mr. Wilson from CSEC Biology TCP. There are two types of immunity: active and passive. Let's look at the animation for active immunity. Active immunity occurs when an individual is exposed to a disease which causes the immune system to produce antibodies specific to that disease. It is important to note here that the individual would have been exposed to the disease, and that immunity is of course being developed as a result of action within that person or organism. So here we have a fellow being introduced to the ranch, and with him he brought pathogen, and you're seeing the pathogen populating the screen in red. And as the pathogen enters the environment, you're seeing that the lady is developing some antibodies that are of course going to be guarding her against this particular pathogen. So of course she will not become ill as a result of her building her defense from within. So here she's exposed to the disease, and as such she would have developed some resistance. With passive immunity, what happens is that the antibodies are of course given to the individual rather than them producing it through their own immune system. So in the passive, you're seeing that it is given to the person, while in the active, it is the person who is actually making the antibody. So in passive, the antibody is made elsewhere and given to the individual. However, in active, it is of course made within the individual. And we're going to continue to reinforce that throughout this video. Here we have examples of passive vaccine. There we're seeing, and of course a mother with the fetus, and we're seeing a mother breastfeeding. Now, immunity could also be considered as natural or artificial. With natural immunity, it is organic. Natural innate active immunity. This is where we start now to think about our exams. Now, these words are all words that they carry specific meaning, and of course direct you to the particular immunity. So natural, it is organic. It has no influence of man. Now, this type of immunity is an immunity that you are born with. How do we know you were born with this immunity? It is innate. That suggests that you were born with the type of immunity. Now, where do we get the antibodies from? Active here tells us that you are making the antibodies. So whenever we see active, it means that the antibodies to safeguard you against that particular pathogen is made within the individual and not with external assistance. Then there's also natural innate passive immunity. And let's decode. Natural, it's organic, it has no human interference. Innate, meaning that it is made within a organism. And of course, passive here mean that it is given. It's something that is given. It is passed from one person to another. When passive, we talk about passive, you are given the antibodies rather than you making the antibody. So here, the offspring gets the immunity from the antibodies that are, of course, produced by mom that crosses the placenta to the fetus. So the antibodies are not made Welcome to TCP Academy, another What's the Difference production. Today we're looking at types of immunity, and of course I am Mr. Wilson from CSEC Biology, TCP. There are two types of immunity: active and passive. Let's look at the animation for active immunity. Active immunity occurs when an individual is exposed to a disease which causes the immune system to produce antibodies specific to that disease. It is important to note here that the individual would have been exposed to the disease, and that immunity is of course being developed as a result of action within that person or organism. So here we have a fellow being introduced to the ranch, and with him he brought pathogen, and you're seeing the pathogen populating the screen in red. And as the pathogen enters the environment, you're seeing that the lady is developing some antibodies that are of course going to be guarding her against this particular pathogen. So of course she will not become ill as a result of her building her defense from within. So here she's exposed to the disease, and as such she would have developed some resistance. With passive immunity, what happens is that the antibodies are of course given to the individual rather than them producing it through their own immune system. So in the passive, you are seeing that it is given to the person, while in the active, it is the person who is actually making the antibody. So in passive, the antibody is made elsewhere and given to the individual. However, in active, it is of course made within the individual. And we're going to continue to reinforce that throughout this video. Here we have examples of passive vaccine. There we're seeing, and of course a mother with the fetus, and we're seeing a mother breastfeeding. Now, immunity could also be considered as natural or artificial. With natural immunity, it is organic. Natural innate active immunity. This is where we start now to think about our exams. Now, these words are all words that they carry specific meaning, and of course direct you to the particular immunity. So natural, it is organic. It has no influence of man. Now, this type of immunity is an immunity that you are born with. How do we know you were born with this immunity? It is innate. That suggests that you were born with the type of immunity. Now, where do we get the antibodies from? Active here tells us that you are making the antibodies. So whenever we see active, it means that the antibodies to safeguard you against that particular pathogen is made within the individual and not with external assistance. Then there's also natural innate passive immunity. And let's decode. Natural, it's organic, it has no human interference. Innate, meaning that it is made within a organism. And of course, passive here mean that it is given. It's something that is given. It is passed from one person to another. When passive, we talk about passive, you are given the antibodies rather than you making the antibody. So here the offspring gets the immunity from the antibodies that are of course produced by mom that crosses the placenta to the fetus. So the antibodies are not made in the baby. The antibodies are actually made in the mother, and of course passes the placenta to the baby. The baby also acquires some amount of immunity from the breast milk. So passive here suggests that you were given the antibodies. It's not that it was made within you. So the baby was given the antibody. It's not a case that the baby is able to make the antibodies for itself. Now innate, of course, when the baby is born, or it is made within. Innate, when the baby is born, will born that with a short-lived immunity, and it is, of course, innate speaks to being within the individual. Why is this youngster running? It seems so happy. Of course, he would have acquired acquired active immunity. What does that mean? Acquired, the immunity that develops after birth. So immunity, if we were supposed to compare, we would compare acquired to innate. So if it's from birth, then it's innate. If it's acquired, then it's after birth. If we were supposed to compare natural, it would be compared with artificial. If we were supposed to compare active, it would have been compared with passive. So this type of immunity is one that develops as a result of exposure to the disease. Example, smallpox and tuberculosis are examples. Now, this youngster probably would have been exposed to the disease, and having exposed to the disease over time, he would have gotten some immunity. So there's a great possibility here that he is now told that he's no longer suffering from the disease. He's immune to the disease because his body would now be making the antibodies to fight off the particular disease. Who wouldn't be happy? Hee-hoo! That's what most persons would be saying. Now, artificial immunity is one that is, of course, introduced, or it has direct human influence. So whenever we go for vaccine, that is a good example of artificial. Now, it could be artificial acquired active immunity. And let's break this down. Artificial meaning that it has some human influence. Acquired meaning that it was, of course, developed after birth. So you were not born with it if it is, of course, acquired. And then active, even though it was given to you by a human, it is within your body that the antibodies are going to be made. So with this type of immunity, artificial acquired active immunity, immunity that develop as a result of individual being given a weakened form of the disease. And the disease here is weakened by eating, or we could use formalin, or we could re-inject several times, and of course weaken the pathogen to a manageable form that can be injected in someone. Usually it is administered through vaccine. Examples would have been that for tuberculosis, polio, and BCG that we would have received in the early years of our life. It's very important for you to pay attention and look at the photographs that we're using in the pictures, as they are giving you prime examples of each of the type of immunity. Now, artificial passive acquired immunity. Artificial here, human influence. Passive meaning that the person was exposed to the antibodies and not to the disease. So here the person would have been given the antibodies because it's passive, and acquired meaning that this resistance, this immunity actually developed after birth. So we have both on the screen, and we continue to emphasize that when you get to the exam, you can break down this long name, artificial passive acquired immunity, what does it mean? And if you were supposed to see passive acquired immunity, what does it mean? And if you were supposed to see passive immunity, what should we understand from this? So here we have broken it all down. We have given it the meaning for all the words in the names, so you should be able to map this nicely when you get to the exam. Now you're seeing this, and this one is synonymous with bites from snake. Now, it is acquired through the introduction of antibodies directly into the individual affected by the disease. And usually you would have seen my first video on serum and plasma. Here we have what is called antiserum, which is used to treat this type of problem. It is fast acting but lasts for a short time, and it is used during cases of emergency where somebody is bitten by a snake or where the person is suffering from tetanus. This type of a vaccine is administered so that there is quick response and the person could of course recover quickly. If you look at this young man, he was bitten by a snake probably, and of course he's been injected probably with of course antiserum to ensure that he will survive. So looking at the snake, it reminds us that it is artificial passive acquired immunity. We want to ensure that we are paying attention and be reminded that you could go over the slide however many times you so desire, so as to make sure that you understand well that which we have covered. We have just looked at what's the difference, and we're looking at type of immunity, one might say types of immunity. I am Mr. Wilson from CSEC Biology TCP for TCP Academy. Please be reminded to like, share, and of course subscribe. And when you do, click on that notification bell and select all, so you'll be notified as soon as there is a new publication. Join us. Today we're looking at types of immunity. And of course, I am Mr. Wilson from CSEC Biology TCP. There are two types of immunity: active and passive. Let's look at the animation for active immunity. Active immunity occurs when an individual is exposed to a disease which causes the immune system to produce antibodies specific to that disease. It is important to note here that the individual would have been exposed to the disease, and that immunity is of course being developed as a result of action within that person or organism. So here we have a fellow being introduced to the ranch, and with him he brought pathogen, and you're seeing the pathogen populating the screen in red. And as the pathogen enters the environment, you're seeing that the lady is developing some antibodies that are of course going to be guarding her against this particular pathogen. So of course she will not become ill as a result of her building her defense from within. So here she's exposed to the disease, and as such she would have developed some resistance. With passive immunity, what happens is that the antibodies are of course given to the individual rather than them producing it through looking at types of immunity. And of course, I am Mr. Wilson from CSEC Biology TCP. There are two types of immunity: active and passive. Let's look at the animation for active immunity. Active immunity occurs when an individual is exposed to a disease which causes the immune system to produce antibodies specific to that disease. It is important to note here that the individual would have been exposed to the disease, and that immunity is of course being developed as a result of action within that person or organism. So here we have a fellow being introduced to the ranch, and with him he brought pathogen, and you're seeing the pathogen populating the screen in red. And as the pathogen enters the environment, you're seeing that the lady is developing some antibodies that are of course going to be guarding her against this particular pathogen. So of course she will not become ill as a result of her building her defense from within. So here she's exposed to the disease, and as such she would have developed some resistance. With passive immunity, what happens is that the antibodies are of course given to the individual rather than them producing it through their own immune system. So in the passive, you're seeing that it is given to the person, while in the active, it is the person who is actually making the antibody. So in passive, the antibody is made elsewhere and given to the individual. However, in active, it is of course made within the individual. And we're going to continue to reinforce that throughout this video. Here we have examples of passive vaccine there we're seeing, and of course a mother with the fetus, and we're seeing a mother breastfeeding. Now, immunity could also be considered as natural or artificial. With natural immunity, it is organic. Natural innate active immunity. This is where we start now to think about our exams. Now, these words are all words that they carry specific meaning and of course direct you to the particular immunity. So natural, it is organic. It has no influence of man. Now, this type of immunity is an immunity that you are born with. How do we know you were born with this immunity? It is innate. That suggests that you were born with the type of immunity. Now, where do we get the antibodies from? Active here tells us that you are making the antibodies. So whenever we see active, it means that the antibodies to safeguard you against that particular pathogen is made within the individual and not with external assistance. Then there's also natural innate passive immunity. And let's decode. Natural, it's organic, it has no human interference. Innate, meaning that it is made within a organism. And of course, passive here means that it is given. It's something that is given. It is passed from one person to another. When passive, we talk about passive, you are given the antibodies rather than you making the antibody. So here, the offspring gets the immunity from the antibodies that are, of course, produced by mom that crosses the placenta to the fetus. So the antibodies are not made in the baby. The antibodies are actually made in the mother, and of course passes the placenta to the baby. The baby also acquires some amount of immunity from the breast milk. So passive here suggests that you were given the antibodies. It's not that it was made within you. So the baby was given the antibody. It's not a case that the baby is able to make the antibodies for itself. Now, innate, of course, when the baby is born, it is made within. Innate, when the baby is born, will born that with a short-lived immunity, and it is, of course, innate speaks to being within the individual. Why is this youngster running? It seems so happy. Of course, he would have acquired natural acquired active immunity. What does that mean? Acquired, the immunity that develops after birth. So immunity, if we were supposed to compare, we'd compare acquired to innate. So if it's from birth, then it's innate. If it's acquired, then it's after birth. If we were supposed to compare natural, it would be compared with artificial. If we were supposed to compare active, it would have been compared with passive. So this type of immunity is one that develops as a result of exposure to the disease. Example, smallpox and tuberculosis are examples. Now, this youngster probably would have been exposed to the disease, and having exposed to the disease over time, he would have gotten some immunity. So there's a great possibility here that he is now told that he's no longer suffering from the disease, he's immune to the disease, because his body would now be making the antibodies to fight off the particular disease. Who wouldn't be happy? Hee-hoo! That's what most persons would be saying. Now, artificial immunity is one that is, of course, introduced, or it has direct human influence. So whenever we go for vaccine, that is a good example of artificial. Now, it could be artificial acquired active immunity. And let's break this down. Artificial meaning that it has some human influence. Acquired meaning that it was, of course, developed after birth. So you were not born with it if it is, of course, acquired. And then active, even though it was given to you by a human, it is within your body that the antibodies are going to be made. So with this type of immunity, artificial acquired active immunity, immunity that developed as a result of individual being given a weakened form of the disease. And the disease here is weakened by eating, or we could use formalin, or we could re-inject several times, and of course weaken the pathogen to a manageable form that can be injected in someone. Usually it is administered through vaccine. Examples would have been that for tuberculosis, polio, and BCG that we would have received in the early years of our life. It's very important for you to pay attention and look at the photographs that we're using in the pictures, as they are giving you prime examples of each of the type of immunity. Now, artificial passive acquired immunity. Artificial here, human influence. Passive meaning that the person was exposed to the antibodies and not to the disease. So here the person would have been given the antibodies because it's passive, and acquired meaning that this resistance, this immunity actually developed after birth. So we have both on the screen, and we continue to emphasize that when you get to the exam, you can break down this long name, artificial passive acquired immunity. What does it mean? And if you were supposed to see passive acquired immunity, what does it mean? And if you were supposed to see passive immunity, what should we understand from this? So here we have broken it all down. We have given you the meaning for all the words in the names. So you should be able to map this nicely when you get to the exam. Now you're seeing this, and this one is synonymous with bites from snake. Now, it is acquired through the introduction of antibodies directly into the individual affected by the disease. And usually you would have seen my first video on serum and plasma. Here we have what is called antiserum, which is used to treat this type of problem. It is fast acting but lasts for a short time, and it is used during cases of emergency where somebody is bitten by a snake, or where the person is suffering from tetanus. This type of a vaccine is administered so that there is quick response and the person could of course recover quickly. If you look at this young man, he was bitten by a snake probably, and of course he's been injected probably with of course anti-serum to ensure that he will survive. So looking at the snake, it reminds us that it is artificial passive acquired immunity. We want to ensure that we are paying attention and be reminded that you could go over the slide however many times you so desire, so as to make sure that you understand well that which we have covered. We have just looked at what's the difference, and we're looking at type of immunity, one might say types of immunity. I am Mr. Wilson from CSEC Biology TCP for TCP Academy. Please be reminded to like, share, and of course subscribe. And when you do, click on that notification bell and select all so you'll be notified as soon as there is a new publication. Join us on a Wednesday, Friday, and Sunday at 5:05 p.m. for live classes streaming from Kingston, Jamaica. Thanks for stopping by. To an interaction between organisms of different species. The interaction may bring. What's the difference? From TCP Academy, I am Mr. Wilson. Today we're going to be looking at symbiosis. Symbiosis refers to an interaction between organisms of different species. The interaction may bring benefit, harm, or no notable advantages nor disadvantages. Mutualism. Both organisms benefit from the interaction. The interaction between a tick and a cattle would have been parasitic. However, the egret removing the ticks from the cattle would have formed a mutualistic relationship in which the egret would have benefited from getting food and the cattle comfort. The sea anemone and the clownfish, as is seen in the picture. Here, the clownfish would have gotten food and protection from the anemone, while the anemone would have gotten nutrient from the dropping of a fish. The fish would also remove organisms that would have, of course, harmed the anemone, which makes it a mutualistic relationship as both organisms are benefiting. Then there is also the nitrogen fixing bacteria that is located in the root nodules of leguminous crop. Here, the bacteria would have gotten somewhere to live while it is fixing nitrogen that the plant is able to use for its growth and other processes. Shark and the remora fish. This is, of course, another mutualistic relationship. Here, the remora fish would have actually hitched a ride on the shark, moving around freely, no energy used, while it would have been eating the scraps left over from the fish, thus providing this clean environment and safe environment for the shark. This is a good example of mutualistic relationship. We're seeing the egret there on the horse, but the egret is popularly found among cattle, and the cattle are standing over there wondering, why is the egret over there and not over here? We have work to be done. We need some ticks to be removed. Predation is popularly called a predator-prey relationship, and it refers to a relationship in which one organism, the predator, hunts and feed on another organism, which is called the prey. So here the shark would have been the predator and man the prey. And there's also this relationship between plants and herbivore, where the herbivore is the predator and of course the plants would have been the prey. Here we have another relationship with the beetle here and the aphid. So the aphid in green, and we refer to this beetle here as a love bug. So this would have been a relationship in which the love bug would be eating the aphid here. A biological control in agriculture. The aphid would be, of course, be parasitic to the plant, as it would have been taking sap from the phloem vessel in the plant. Commensalism. Commensalism refers to a relationship in which one organism benefits and the other is not affected, as in the case of a hermit crab using the shell of a dead snail. The snail is not affected, but of course the hermit crab would have gotten protection from the shell. Another example here is the egret, which follows behind a grazing cattle. Now, in this particular relationship between the cattle and the egret is not really feeding from the cattle. As the cattle moves through the grass, the egret would, of course, feed on whatever is found in the grass and not on the cattle. Here, the egret is benefiting, but the cattle is in no way affected. Then there is, of course, the epiphytes on trees. And epiphytes would be like this plant here growing on another tree. It is there getting sunlight, but it is not affecting the plant in any way, shape, or form. So it benefits from this high elevation. No predator would be affecting the red plant here seen on this tree. However, it would have been able to carry out photosynthesis without hurting the plant on which it is perched. This is a popular one, parasitism, and this pretty much refers to a relationship in which an organism is of course hurt while the other organism is benefiting. So here the mosquito is a parasite to human here having a grand time, as they are of course sucking the blood from the human and could very well introduce the plasmodium, or one would say could cause dengue or malaria. Now the mosquito is harming man. Tapeworm and man is also another example of a parasitic relationship, and of course the ticks on the cattle or on human is a good example of a parasitic relationship. Here we have a ticks, and this is really dangerous to both cattle and other organisms. Amensalism, this is not on the CXC syllabus, but it refers to a relationship in which one organism is hurt or destroyed while the other is unaffected by the interaction. So the elephant here moving through the grass is of course affected. The elephant is not affected in any way, as it just moved pretty much through the grass. Penicillin are pretty much killing a bacterium. Here this is a case where the penicillin is not affected, as penicillin is pretty much from a fungus. So the fungus is not affected, but the bacteria is of course killed. And another thing we could look at is competition. Here we could have intraspecific and interspecific competition, where intra speaks to a competition within the species and inter with different species. And then we have antibiosis. Antibiosis speak to a relationship in which one organism secretes a chemical to kill another. The last one we look at here is mutualism, and this is not on the syllabus for 6C. Here, organisms, they are usually found together, but they benefit nor affect each other. They are just there, as we would find plants in the desert. This takes us to the end of the presentation for today. And of course, please be reminded to like, share, and of course, subscribe. Also, be reminded that you can benefit from our many past papers on the channel. of immunity, and of course, I am Mr. Wilson from CSET Biology, TCP. There are two types of immunity: active and passive. Let's look at the animation for active immunity. Active immunity occurs when an individual is exposed to a disease which causes the immune system to produce antibodies specific to that disease. It is important to note here that the individual would have been exposed to the disease, and that immunity is of course being developed as a result of action within that person or organism. So here we have a fellow being introduced to the ranch, and with him he brought pathogen, and you're seeing the pathogen populating the screen in red. And as the pathogen enters the environment, you're seeing that the lady is developing some antibodies that are of course going to be guarding her against this. Biology, the cover page. If you will be doing a biology or human and social biology exam and genetics is your concern, worry no more. Stay with me as I work out and explain Punnett square, genetic cross, and sex-linked inheritance. What is sex-linked inheritance? It refers to a condition or genes that is carried on the sex chromosome. In males, the sex chromosomes are X and Y, while in females, the sex chromosomes are XX. It is always possible for both male and female to inherit the X-linked trait, as they both have X chromosomes. Y-linked traits are, however, confined to males since they are the sole carrier of Y chromosomes. Recessive traits that are associated with the X chromosomes only affect female if it is inherited in the homozygous recessive form or genotype. If a trait for the recessive allele is found only on one of the X chromosomes in women, they are not affected by the trait, as the condition trait is usually masked by the dominant allele. In such case, the woman is called a carrier and is able to pass on the recessive allele to her sons. Even if a father did not have the trait, she will also be able to pass on the trait to half of her girls. Her daughters, however, will also be carriers. The male is not able to inherit two recessive alleles. This is due to the size and type of chromosomes, XY. However, if they inherited one of a recessive allele, they would be affected by the trait, as the X chromosome is taller than the Y chromosome. Hence, there will be no dominant allele to mask the recessive trait. Here we have a diagram of the chromosomes, X and Y, male and female, and we're looking at common H here being the recessive allele and capital H being the dominant allele. Now what we're saying, it is on the X chromosome that the trait would be. Now in the case of the male, as such, once the male has the recessive allele on the X chromosome, the male will be affected by the trait. On the other hand, the female here has one recessive allele and a dominant allele as well. In this case, the recessive allele is masked by the dominant allele capital H here. The capital H is the dominant allele. As such, it will mask the common H, which is the recessive allele here, hence making this female a carrier. Now, a carrier female is not adversely affected by the trait. However, she will pass on the trait to her daughters, but her daughters will only be carriers. Hemophilia. This is a disease in which the blood fails to clot. Hence, even with small cuts, the individual bleeds more than normal. Allele capital H is the normal clotting allele or dominant allele, while allele common H is the recessive allele for the trait. Color blindness or red-green color blindness. In this case, individuals suffer from this condition are not able to see some colors. The condition is observed with red, green, and blue. This can cause major problems with indicating lights, colors, and the perception of fruit maturity. Let's look at an example of the genetic cross or the genetic diagram as we would have it. First, we're defining the allele here. So the first individual is a female and she has the allele for normal blood clotting. The second individual is a carrier female. She has the recessive allele, over its masked by the dominant allele, and the dominant allele is the capital H for those persons who are just starting genetics. Now, this person will not be adversely affected by hemophilia. However, this individual will be able to pass on the hemophilia to the F1 generation. The third female is a female that is affected by hemophilia. She's a hemophilic, which means that, of course, she is adversely affected by the condition, and so too she'll be able to pass on the condition to her offspring. The other individual is a male, and this is a male for normal blood clotting, as this male has the dominant allele H, which will cause this individual to have normal blood clotting. The last male is a male that suffers from the condition hemophilia, so we say that he's hemophilic, as he has the recessive allele for hemophilia. Now that these are well defined, let us look at doing a genetic cross, which is well animated. At any point you can stop, replay the video to ensure that you have a good grasp of doing the genetic cross. So we start by looking at the question. The question we have here is that a normal male is crossed with a carrier female for hemophilia. We're supposed to determine the phenotypic ratio of the F1 generation. We're supposed to show all steps as we complete the genetic diagrams. So here we go. We know that the genotype for the individual must be written, and we know the phenotype, of course, for the two individuals. One, of course, is a normal male, and the other is a hemophiliac or hemophilic carrier female. So here we have just the male female as phenotype, nothing attached there. However, the female should have been written there as a carrier, female carrier. Now the genotype is well written. It's a normal male, and you're seeing there the dominant allele, capital H on the X for the male, so he's not affected by the disease. However, the female here, which is a carrier, and the phenotype I said before should have been female carrier. However, the genotype is correct. The genotype for the carrier female is a dominant allele, capital H, a recessive allele, common H, and they are both on the X chromosome. Now, the next thing we are going to do, we are going to be identifying the gamete. So here we go, the gamete with the allele there. Good. So that is your first step when you get into the exam. You're going to be identifying the phenotype from your question. You're going to be identifying the genotype based on what was given to you, and then you are going to break away with the gamete showing the chromosome and allele, and of course then we're going to be moving into segregation, or what we call random fertilization. Now look what happens here. So we're trying to identify the F1 genotype. So that is the first part of the cross, and here we go with the second. So observe, they are color coded just to show you where the chromosome, the allele, where they are coming from to make the F1 generation or the offspring. Right, there we go. Now please make note of this as we move to the other side. So there we go. Just let me pause and identify this bit. If you are observing, here we have two arrows with two chromosomes. So here this is completed. Here we have two arrows with two chromosomes. Here this is completed, right? We have one here, which means that we need to have another coming from it. We have one here, which means we need to have another coming from it. Same too for this chromosome. Now this here is completed. There's nothing else that can come from this because we are doing monohybrid inheritance. All right, we're just looking at one trait. So here we go. Now completed, and we have just one other. All right, so if you look here, completed, 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 but this here still needs another arrow, and this here still needs another arrow. Here we go. One down. So here it is completed here. So now this needs another arrow, and of course this needs an arrow just the same. All right, so we're going to be crossing from here to here with that arrow. Here we go. All right, that completes the genetic cross, and exactly how you're supposed to be doing it for your CSEC examination. Remember, at the top here, you must be paying attention. At the top here, it should have been female carrier. Female carrier. We only have female there, but it should have been female carrier. Please ensure that you make that correction. Now we're supposed to be identifying the phenotype, but we could identify everything here before. Here we have the phenotype, which is taken from the question, and the genotype. Based on our interpretation and our prior knowledge, we know that this is what it should be. And then we come down to the gametes. Here we have random fertilization here, or some might say segregation. And then we come to the F1 generation, which stands for the first filial generation, the first set of offspring from these parents. Then now we need to identify what these offsprings are. Are they affected? Are they male? Are they female? That type of thing we need to identify. So let's look at the phenotype. So here we go. So the phenotype for the first individual, it's a female normal, which means that she is normal for blood clotting. Her blood will clot perfectly well. Then the other, we have a male, is a hemophilic male, as he has one recessive allele, and of course the only allele that he could have had, since the allele is linked to the X chromosome. Then we look here, we have a normal male, since he has a dominant allele H linked to the X chromosome. Here we go again. And the final, we have a female carrier. And if you remember when we started out, we said this is what it should have been, a female Can you hear me now? All right, great. So you should be hearing me now. Are you hearing? Just indicate in the chat for me. Good morning and welcome to the early show with Mr. Wilson. We are continuing our preparation for the CSET biology examination and HSB examination. The biology exam is slated for Monday, while the HSB is slated for Wednesday. Our preparation, though it be, or as it is now, the biology paper, it does not mean that questions on the biology paper will not show on the HSB paper. It doesn't mean that. From my knowledge, I have seen in many instances where the exact questions are on both papers. So we want to ensure that we are following that lead. And those persons who are doing HSB, you want to log on to make sure that you're a part of the wonderful offer. Now, this morning we're going to be looking at the 2020 paper. If time allow me today, I will make available. We would have done 2019, 2017, 2020. So we have the 2021 and 2018 paper that should be in your possession by tomorrow. We're really, really grateful that you have joined. Just tell us in the chat, where are you joining from, which Caribbean island, or if you're joining from Jamaica, would love to know that. We always want to hear where our viewers are from so that we better understand the need of our people and we can do that type of a delivery. Now, while you type in the chat where you are from, I started looking at some possibilities, as it were, based on the trend that we have been seeing with the exam. So I think I want to call some questions for the paper. Be reminded that I am not an employee of the CXC. I don't know what is on the paper. This is just a mere assumption based on trend. So I think that the carbon cycle and decomposers will be on the paper. Based on the trend, genetics will be on the paper. Based on the trend, disease is going to be on the paper. Birth control is going to be on the paper. A reproductive system or the menstrual cycle, I am thinking that that's going to be on the paper. Evolution and heredity, which of course genetics is a part of, I am almost convinced that those are going to be on the paper. Now, be reminded that while I would have said this or made this declaration, I am not an employee of the CXC and I have not seen the exam paper, so I can't say for sure whether or not these will be on the paper. It is just my guess as to what will be there. So great. So persons are hearing. So let me just pop on the screen. So you'll see yourself coming on the screen. So here we have from Jamaica. We have Carlene from Jamaica. St. Kitts and Nevis. We have Janelle from St. Kitts and Nevis. No, Janelle is from Jamaica. So we have from St. Kitts and Nevis, that I think that's Jermaine. All right, St. Kitts and Nevis. So three persons of a lot would have shared with us their country. As they come in, we will look for that. Oh, we have somebody from Jamaica here. We have Lee here from British Lee from Jamaica. I think I saw something in the post from British Lee. I think I did respond. All right, so that is pretty much a compliment. We are going to just get going with what we have. We can't spend the entire time. Now, pretty much the preparation for exam is pretty much a student ordeal. I really facilitate that conversation and assist where I can. So it's pretty much a student-led initiative. So I'm going to just hop right into the past paper as it should be. Let's just look at that. All right, great. All right, so we have Stacey Ann, and Stacey Ann is from Jamaica. She's on screen now. So we're looking at the 2020 paper. And of course, it is the January paper. You would imagine, because COVID came, I think, somewhere in March, so there was no CXC, I think, for that year. And it is, of course, a paper two. I like to say to my students that you are supposed to read everything on the paper. Just let me get all these things off the screen. All right. All right, great. I like to say to my students that you are supposed to read the entire paper to ensure that you are following what the instructions, or you understand pretty clearly what you're supposed to do. So most time persons ignore this part of the paper. You want to ensure that you are reading and you know your rights in the examination. So we're going to move on. And one thing with this paper, I think this paper is pretty similar to a paper I worked yesterday. Could have been the 2017 paper. I think on the 2017 paper, there was something about transpiration. I don't remember, but my mind is telling me something like that. So this is the first question for us to answer. And it reads, and I'll make it a little bigger on the screen. It reads, describe the process of transpiration in plant. Your description must include the two main, should include the name of two of the organs involved in transpiration. So tell me what you have for this in the chat. And the value of this question is, of course, 4 marks. So what is required? You're supposed to briefly describe the process of transpiration in plants, and your description must include the names of 2 of the organs involved in transpiration. Let me see what you have there in the chat as we move with the early show, trying to establish a part for the CSEC examination. I know this morning the internet is going a little slow, but of course we're going to make it through whichever way. All right, so we want to answer the question that is on screen. Just type it in the chat as much as you can, and I'll make the correction as much as is needed. So we are looking at transpiration, and you are watching CSEC Biology, TCP, with Mr. Wilson, or the program is, of course, Mr. Wilson Live. I do hope that the transmission is reaching you and you would have heard the question. So we want to look at transpiration. The first thing we think about transpiration is that transpiration is a process by which water leaves the leaf, or process by which water evaporates from the plant. That is something that we want to have in our mind. Now, 2 of the main organs that are involved would have been the root and, of course, the leaf. As the water would have entered the root via osmosis, and then the water would have moved up through the xylem vessel by transpirational pull. As water leaves the plant, the cohesion causes the water to be pulled up, and of course it leaves the plant via the stomata. So that is something that we want to look at and be able to talk about in the exam. Let's look at the other question. The other question wants us to look at this particular experiment. And for most persons, you would not have done such an experiment at school. An experiment was carried out to determine the effect of relative humidity on the rate of transpiration. The results are presented in the table. Use the grid on page 5 to draw a graph to represent this data. Now here, if you observe, we have table, and the table is pretty much the effect of relative humidity on the rate of transpiration. Now, if we're going to be drawing a graph, the graph will need, of course, a title. So we could pretty much put graph and then use the same title as we move on down to the graph leaf to put that information on the graph leaf. So we would put the title here. It is requesting that you put the title there. And as much as possible, if you can write it in uppercase, that would have been nice. Now I'm going to just minimize the screen one bit in order to solve this question. Now we're going to be putting this information on the graph. Now, if you have watched my previous lives or my previous lessons or my previous past paper that I have done with graph, I would have pretty much indicated what information is placed where on the graph. Now let us be reminded that it is the independent variable that is going on the x-axis, and usually the independent variable is the first column of your table. So here relative humidity will be placed on the x-axis, and the rate of transpiration, which here is measured in milligram per centimeter square for the given time. So we're going to put this on the graph, but we're going to be having one problem. Now, all graphs should have a scale. So we want to work out a scale. Most person would be familiar with a scale, and that pretty much represents how the information will be represented on the grid that we have. Now, what I did prior to this show, if we're looking at the x-axis, I said that 1 centimeter on the x-axis would have been equivalent to, I think I said 5. 1 centimeter would have been equivalent to 10. Sorry. 1 centimeter would have been equivalent to 10. 10%, that is. Here we have a percent. So 1 centimeter would have been equivalent to 10%, right? And on the y-axis, I think I said that 2 centimeter would have represented, I'm doing the scale here, 2 centimeter would have represented 5 milligram per centimeter square. That pretty much would have represented the rate. That would have represented 5 there. Now, having worked out the scale, the other thing that we need to do is to just move on down and start drawing the thing. So you're going to write a title here. I remember that we said that the title will come from the graph. We're going to be using the same title from the graph in order to write the same title from the table in order to write the title for the graph. So you want to make sure that you can do that. So just move this and put it on top of the graph, and you should be fine. Now here we want to draw the axis for the table. Yes, there's a school of thought that said about 75% of the graph leaf should be used. But there's another thing that an intelligent man tend to manipulate his environment. So you want to make sure that you are manipulating your environment, that it is working for you. So let us think about drawing this. We could start drawing on the inside here, or we could start drawing on the outside, whichever. So you're going to draw your graph using a ruler. I'm not going to draw it there, but you just use a ruler to draw it and not just to start numbering on the grid. Now, please be reminded that your graph should have directional lines. So when we talk about directional lines, it should show the path in which the variables are increasing. So we're increasing in that direction, we're increasing in that direction. Another thing that it needs to have is the name of the axis. Now, the name of the axis will come from the table here. So relative humidity, RH, will be on the x-axis. So RH is going to be on your x-axis. Relative humidity is the independent variable here, RH. And on the y-axis, we are going to be having a rate of transpiration, and we could write the unit in which it is measured. So relative humidity here is measured in percentage. So you just put that there in a bracket. And then on this side, we are looking at the rate of transpiration, and you would have seen the unit on the paper in which the rate of transpiration is measured. There we have it. Rate of transpiration, milligram per centimeter square per hour, right? And then all that we need to do now is start populating the graph. Now, based on our workout, we could use this as 10, this was 20, 30, 40, 50, 60, 70. I'm using my touchpad, so forgive me. 80, and then 90, and then we would get up to 100. All right, just bear with me. All right, so I've gone up to 100. Yes, somebody might argue that we have not used up the 75% of the graph, but look at what we're going to do. and then we can say if we did well or not. So I want to start going up here by 5, and we're going to go up here by 10, and then we go up here by 15. So we're increasing in increments of 5, and we get here for 20, and then we go 25, and you would have realized that the graph pretty much stops at 25. But by the time you are supposed to draw your scale in, by the time you draw your scale in, you would have realized that a significant portion of your graph paper would have been all used up, and it looks pretty much like 75%. So you just draw your scale in there, what the x-axis should be here, and the y-axis, and you want to label it that it is a scale. All right, so you want to label that as a scale, and then you want to put, like I did earlier, 1 centimeter equal whatever. The thing I did up at top, you want to get this information here, get this information onto the graph here, so it's a scale. Great. Now the next thing that we need to do, we need to plot or place this information onto the graph. So from the table, we know that at 10%, we had a 25 millimeter per, the rate of transpiration was 25. So all that we need to do is to come onto the graph, look at where it intercepts, and we are going to be plotting there. So I'm using some X right in the intersection there, and the same is true here, and the same is also true here, right? And then we are going to continue plotting. I think after 25, 25, we have 20, 15, 12, 10. So we have 20, 15, and then if this is 10, this is going to be 12 and a half. So it is somewhere about here. All right, let's move on back up. 12, then we are going to have 10, 8, 5, 5. So we have 10, and then if we have 10, then 8 is going to be somewhere here, and then we are going to have 5 here, we're going to have another 5 here. All right, so realize that's a line graph. So we're going to be drawing this in. So we're going to come right across. We are seeing some constant here, and then we are going to swing this down and swing this line right through this area. I don't think I'm doing so bad using the touchpad. And then here we'll see a little plateau becomes flat right there. All right, so that pretty much gives us the graph that we are looking for. All right, so now it is time for us to answer question. Let me come on over and see what's happening. My friend from St. Kitts, how is that graph? Is everybody comfortable with that graph? It was easy, just like taking out the information off the table and putting it on the graph. Right? So now that we have done that, the next thing that we need to do, so don't run away from your graph questions. Check out my mini graphs that I would have done, and you should be fine. All right, let us look at the other question. Now here it is saying use the result from the table table 1. State how the rate of transpiration changes as the relative humidity increases. So as the relative humidity increases, right? Look at it and say how the rate of transpiration. So it says using the table, state how the rate of transpiration changes as the relative humidity changes. So we are looking at how the rate of transpiration, what happened to this as this change, as the independent variable change, what happened to the rate of transpiration. Tell me in the chat and let me see if you have a good grasp of that. What happened to the rate of transpiration as relative humidity changed? Tell me in the chat. As relative humidity changed. Okay. Anybody else? I really needed that answer. Anybody else? All right, so I will go with Rajay. All right, Rajay, it is always going to be the easiest answer, but I'm going to take you back to the graph and I'm going to show you why that couldn't be the best answer because it is not totally true. So let me just change my ink and let me show you something here. Now, if you observe here, just let me look at your answer again. You are saying increase. No, I was expecting you to say decrease. It's not increase or decrease. Transpiration increases. No. No. As relative humidity increases, transpiration rate decrease. If it is coming down, it's going to be decrease. If it is going up, it is going. So remember, once a line on the graph, just let me show you this. And I've done this a couple of times well now. If a line on the graph is going in that way, that is increase. If a line on the graph is going this way, that's a decrease. All right, so you need to know those stuff. This way, A, that is an increase, and B, that's decrease. So if it's coming down here or slant like this, it's a decrease. If it's going up like that, it is an increase. Look at the direction. For those persons who do agriculture, econ, under the law of supply and demand, you know about these curves. All right, so I wanted to point out why that decrease answer would not have been the best answer. All right, so relative humidity increases, transpiration decreases. All right, great. So let us look at this answer, which is the closest to being plausible. And I'm going to explain why that can't be true and why we couldn't answer it that way to get full marks. If you observe between 10 and 30, humidity is increasing. All right. But would you say the rate of transpiration is increasing here? Look at this part of the graph. The rate of transpiration is not increasing there. And if you look at this, it's not decreasing there. Now, if you look at this part here as well, humidity is increasing, but the rate of transpiration is not changing here. So because of these two parts on this curve, then that would not have been the most suitable answer for your graph. So you want to try again to see what would have been the best answer. The thing about this, you would have to apply some scientific knowledge as to how to word this particular answer for this problem. All right, so let us go back. Do you agree with me that the answer couldn't be as is on screen? It couldn't have been. All right, so I'm going to help you with the wording and probably you can write it. All right, so as the humidity increased, are you with me? And the concentration gradient between the water in the leaf and the water outside of the leaf. So let me do that again for you. As the humidity increases, and the concentration gradient between the water in the leaf and outside of the leaf as humidity increases and the concentration gradient between the water in the leaf and outside of the leaf, as that concentration gradient become low, the rate of transpiration decrease. So it is about the concentration gradient of the amount of water in the air versus the amount of water in the leaf. Now, when they are somewhat, should I use isotonic? When they are somewhat equal, that is when you are seeing no much movement. But when there is a gradient, when one is higher than the other, then you are going to see a movement. So as the case that you have there on the screen, it is as a result of that concentration between the leaf. And of course, when that becomes low, then what happens is that you are going to be having changes in the rate of transpiration, and that is what is going to drive the graph looking like this. So we could not say as the rate of transpiration increases, we couldn't have said that, because if we said that, those two parts that are circled red would have been incorrect. Instead, it is as the rate of humidity increases and the concentration gradient between water and the leaf and outside of the leaf, as that becomes low, the rate of transpiration will decrease. All right, so that's pretty much the answer for this particular question. You probably could find something like this in your textbook if you were supposed to go to transpiration and look at humidity. All right, so this is sort of a trick one from CXC. I'm thinking that they did you wrong there, because what they did was for you to just quickly look at this diagonal line and not paying much attention to the area that is circled in red. You want to pay attention to that. All right, so it is about the concentration gradient. As the concentration gradient changes, then that is what would cause, that would have been your answer. All right, so the other thing we're going to be looking at, the other question is going to be suggest two ways by which the result given in the table support your answer in 3. So if you observe here again, they are giving you another chance to look at what you wrote, Rajay, to see if that answer is correct. And if you wrote the wrong answer, having seen this question, you would have realized, whoa, I wrote something incorrect. It's time for me to change it. So if you were supposed to look at two ways in which the result given in the table is supported by your answer. One, you could say between 10 and 20% humidity, the rate of transpiration was 11. It was 11? Was between 10 and 30 percent humidity, the rate of transpiration was 25. So that's where we have that first flat area at the top. Now, as the concentration gradient change, you would have seen that diagonal slope. Now, as the concentration changed between 10 and between 30 and 90, then the rate of transpiration also slowed. I don't know if you got that. So let me just go back to the thing. We want to support our answer here. So we are saying between 10% and 30%, the rate of transpiration was 25, right? So that is why we had to use the answer that we use. However, between 30 and 90, the rate of transpiration slowed. All right, what you have seen here is that the rate of transpiration would have fallen. So this is what support our answer. So our answer there is to say that between 10 and 30% humidity, the rate of transpiration was, of course, 25. However, as the concentration changed between 30% and 90%, the rate of transpiration slowed. So you would have realized that transpiration is taking place much slower. All right. I know it started to sound a little tricky to a lot of persons who probably would not have read much on transpiration. Next thing, a farmer observed that his plants wilt during the day but recovered at night. So just one reason for this occurrence. So write that in the chat for me. All right, try this one for me in the chat. One reason for this occurrence. So the plant is wilting during the day, however, it returns to its flush status at night. What is your reason for this? What's your reason for that observation? Write it in the chat for me. So the farmer would have realized, there's a question on screen. A farmer observed that his plants wilt during the day but recovered at night. Suggests to the farmer one reason for this occurrence. What do you think is the reason for it? Okay, let me read. Phillips supplies. I am not too sure with supply. I'm not too sure what oxygen. I'm not too sure what you're answering there. Plant is dehydrated in the day. Okay, so dehydrated meaning that there's not enough water in the soil. But what happens for the night? You told us about the day. What about the night? So during the day, temperature is usually high. So therefore, the guard cell will close, not allowing water to leave. Now that is true. Not that is true. If it's losing too much water, that will definitely happen. But what happens during the night? All right. During the day, the temperature, let me just share this with everyone. Not too sure what it is saying, but I'll share it on screen so you can see. So it says that during the day, the temperature is higher. The transpiration rate are higher. However, during the night, the temperature is low, hence transpiration rate has decreased. Okay, this is sort of plausible somewhat, and I've seen others that are somewhat similar. All right, so the answer is that there's but little water in the soil during the day. As soon as transpiration rate increases, the amount of water in the soil would have been depleted. So the cells, of course, will become flaccid. When the cells become flaccid, it's as a result of losing water. So in that case, the cells tend to become, having been flaccid, then of course the plant will show signs of wilting. However, at night, less water is lost from the plant, so the soil will pretty much tend to have a little more water from dew and all of that being on the soil. The soil will have a little more water, so the plant will, of course, return to its flush condition or somewhat turgid. All right, so let me see. Water loss due to, let me see this one on the screen. Water loss due to photosynthesis in the day, while at night there is no photosynthesis occurring, no sunlight present. All right, great. So always be reminded that the plant wilting, it is as a result of losing too much water. Now, once a plant is losing too much water, what will happen naturally is that the plant will close the stomata so that there is no water leaving in order to protect itself. However, during this time, the plant will experience what we refer to as transipient wilting. Once a plant enters transipient wilting, then what will happen is that it waits for water to recover. And if there's no water to recover, then it goes into permanent wilting and will definitely die. However, in this case, the plant will find some amount of water in the soil based on the environmental condition or the farmer water in the soil. And of course, the plant will return to its nice flush condition. Great. We had some great attempts there. All right, so just two ways in which animals transport system and the plant transport system are different. So I want to look at two ways in which the plant transport system and the animal transport system is different. So just two ways in which the plant transport system and the animal transport system is different. So by this, we would have known that transport will be on the exam, and transport, you are looking at both plants and animal. So how is it that the plant transport system versus the animal transport system, how is it that they are different? What makes them different? Tell me in the chat. As you type in the chat, I think I want to call some of the topic that might be on your exam, owing to the fact that I don't work with the CXC. These are just my opinion. I strongly think that the carbon cycle and decomposers will be there. Come to think of it, decomposers must be there because the syllabus said decomposers, and there are no other way to put decomposer than decomposer. They are always associated with the carbon cycle. Among other things, if we look at nutrition, decomposers are also associated with nutrition. So we are sure that decomposers will be there. However, we think that the others might be there based on trend. So we think that the carbon cycle will be there, decomposer will be there, genetics, which is a part of evolution, and heredity, that of course will be there based on the trend. There will be a genetic question, evolution, heredity. We're thinking that diseases will be there. Based on the trend, birth control is going to be there. Based on the trend, the reproductive system should also be featured in this exam. Those are just our assumption. We don't work with the CXC, but we strongly think that those are some topics that we could see on the paper. All right, so plants are autotrophic while animals are heterotrophic. I think, Peter, you would have misinterpreted this question. Let me look at this one on screen. We're looking at the transport system now. Transport system. So just let me share this on the screen. It says here, animal transport system comprising of blood vessels, which are arteries, veins, and capillary. That's a good move. Whereas in the plant, substances are transported in the xylem and phloem vessel. Great. But it asks for, was it two that it had asked for? Just let me check. This is a great answer though, but I think that it had asked for two. So you have just provided one. So what is the other? Remember, if we're asking to suggest two ways that they are different, that is just one comparison. Think about using a table. If we're using a table and we had the 4 animal and the 4 plant, we would have just done one comparison. So we need another one now. We need number 2. So what is number 2? The answer you gave just now, that's a wonderful answer. So that could be your wonderful number 1 or your number 2. So what is the other answer that we're going to have here, girl? We need another answer. Now, blood is transported. Let us look at this one from Charles. I'm going to share it on screen. Blood is transport. Blood is the transport medium for animal, while plant transport medium is mainly water. I wasn't looking at that, but it is somewhat plausible. I was looking for persons to take the easy part, and the easy part is going to be that the animal, in most cases, as we're looking at this, let me make sure that I'm going to tell you the right thing now. Let me see the question again. So just 2 ways in which animals transport system. Okay, so I'm thinking about animal here being like multicellular animals, and probably I'll just write multicellular animal when I'm answering the question. So I was thinking about they have a heart that pretty much would pump the blood around the body, while for the plant, it does not have the heart to do that. It relies on capillarity, it relies on transpirational pull, relies on turbo pressure. That type of thing move the substances in the plant, while the body, the animal body, would definitely use a heart. But I have seen interesting questions, interesting answers, and that is why I like to hear from students, because they are always showing you a different side of a coin. Be that you got it right or got it incorrect, the wrong answers are also needed. so persons will understand that these are not answers that are accepted for the particular question. So don't feel too bad if you didn't come in on board. Now, the transport system in animal transport oxygen, etc., while in plant transport system. Okay, just let us be reminded that in the plant, just like in all other organisms, oxygen moves through the plant pretty much just the same way because the plant carry out respiration. So Phillips would have to revise that answer there. It might not be so close to what we're looking for, because it is true that the plant also uses oxygen and it has to get to the various parts to carry out respiration. At this rate, you feel like you're failing. I'm not too sure why you feel that way. I sound okay. All right, great. So let's move on. So this thing differentiate between the transport need for a unicellular animal and a multicellular animal. The transport need for a unicellular animal versus a multicellular animal. What the transport need? Tell us the difference there in the transport need. What's the transport need? Anybody can answer the question? Why do you feel that you are failing, Phillips? The exam is still in front of you. Anybody can answer that question? We are looking at the need for a transport system in multicellular organism versus unicellular organism. No one can? Okay, great. So with the multicellular organism, they have a very small surface area to its volume ratio. As such, they need something like a heart in order to pump the blood around the body. In unicellular organisms like the amoeba, they have a very large surface area to their volume ratio. And as a result, sir, wait. Okay, I'll wait. So that is pretty much the answer for that question. And for persons who are seeing some of these for the first time, remember that we went for some papers that we think would have scored high for the exam. So don't worry yourself. All you need to do is to familiarize yourself with the questions and, of course, the topics that we're looking at for the exam and be exam ready. So we are here different. We want to differentiate between the transport need of unicellular animals and multicellular animals. What is your answer? I started answering for you. So, okay, great. So I remember you always can correct Mr. Wilson. I don't mind that. So it is not about the organism being big or small. You have to speak, use the language of the syllabus. So multicellular organism like you and I, we have a small surface area to our volume ratio. And as a result, we need a heart. If I mix it up, just remind me. And for the amoeba, they have a large surface area to their volume ratio. And as a result, they wouldn't need like a heart to pump or whatever they need around the body. They could just carry this out by simple diffusion. And this is a question that is always on the multiple choice paper. So whichever way it is coined, you want to remember, it is always, this particular question is always on the multiple choice paper. Now, both plants and animals need to store food. With reference to one storage site in animal, explain why food storage is important. So that is something that is popular in your textbook. Why is the storage site important for organisms? With reference to an animal. So if you're going to make reference to the animal, you need to speak about the storage site in the animal, one, and two, you need to tell why it needs to store the food. So pretty much if we are looking at the animal, we did this yesterday or last night at length, where we looked at the muscle and the liver. We spoke about glucose being converted to glycogen and stored, and when we needed it, glucagon would have converted it back into glucose. I didn't tell you last night that there's also adrenaline that will also quickly convert the stored energy to energy that is needed to be used. So adrenaline is another hormone there that works to convert the glycogen back into glucose. And we are going to need this for time when there's scarcity in energy, or need it in time when there is an emergency need for energy. For example, you will recall that the seed is storing energy in the cotyledon, so that at the time of germination, when no photosynthesis is taking place, the seed would have had this energy in order to carry through germination. The same is true. I am seated here, but I'm not connected to the electric plug to get energy to use. So I have to have energy stored like a battery so that I can move about and do what I have to do on a daily basis. So pretty much I would have to have had as energy storage in order for me to do that, or else you understand I would have to just be plugged in or have a gas tank or something like that providing the energy. So I store that energy in order to do my everyday chore. I store that energy to go through need times of scarcity. I store that energy for reproduction. I store that energy for locomotion. I store that energy just to do everything that I need to do. Good morning, Pinto, and welcome to the show. All right, so we would have just completed question number 1. Now, you would have realized that though we did transpiration, and we have been looking at transpiration about 3 papers now, there are still some persons, depending on how the question is asked, they're a little shaky. Now, we are moving on to a new question, which is question number 2. And question number 2, in my opinion, is a little easy question, but I must point out some of the ills that CXC have on this paper. When you are drawing and labeling, you should not use arrowheads. All right, this is wrong. All of these arrowheads something, they are wrong. I don't know why CXC keep carrying things like this on the paper. I don't know if they try. I don't know what they're doing. I don't know the intent. I'm not an examiner. I'm not so bright like those persons. All right, so figure 1 show the internal structure of a generalized plant cell. Now, we're looking at the generalized plant cell here, and we want to identify the parts that are labeled. So I want you to tell me in the chat what are the labeled parts. So we talk about the adipose tissue. Yes, that's an example. It's converted and stored. Fat is stored around organs and under the skin. So that is also a very good move there, Charles. Very good move. Just let me show what you have on the screen for the question we answered just now. So it's saying the adipose tissue in animals store fats. These store fats are used for insulation as well as to be used when glucose is scarce in the body. Yes, that's a good one. Let me see what Philip is saying. All right, fats are one of the storage site. Fats are storage site in animal, and in case where there is a scarcity, the body will convert the fat into energy. Well, that is not bad, just that we need to, there are some little things that we need to shape up, but that's a good attempt. All right. All right, so persons are answering what's on screen. So Tori is saying that A is the cell membrane, and she says that B is the cell wall. I might need to just increase this here, that persons can see it a little better. All right, I do hope that that can help. So this is probably the easiest thing in biology. Everyone should be able to answer questions surrounding cells. So just let me erase that which I have there while I wait on you to answer this question. I don't think you need my help in answering this question. I really don't. All right, so C, cytoplasm. Somebody said D, vacuole. All right, so I'm just going to pop some of the things on the screen. All right, so I'm seeing Philip is saying B, cell membrane. Also, B there should be the cell wall. Let me take that down. C, cytoplasm. This one is really interesting. Look at this one here, F. I think that's going to be the hardest one. So if F is the chloroplast, yeah, and E is, of course, the nucleus. So let me just put the answers on screen based on what you have. So F there being the chloroplast, E there being the nucleus, C, cytoplasm, let's move on up. I'm just going to be using that which you have here. We have a B is going to be the cell wall. And let me go on up to see if there's an A. D is going to be a vacuole. And we have an A, which is, of course, going to be the cell membrane. Beautiful. But I want to ask you a question. I want you to look at, I'm going to circle it. What is this? This organelle here, what it is? The one I circle with red, tell me in the chat what it is. The organelle that has been circled with red, what it is? Okay, great. So the organelle that is circled there, it is the mitochondria. And one of the things for those persons who are not identifying these things well, just remember that the mitochondria tend to have this thing like an invagination, while the chloroplast is pretty much a stock of cristae. All right, something more like that, right? While the mitochondria tend to have some invagination as it is here. And this here, what is this organelle? Anybody can tell me what that organelle there is? See, I'm touching it here. What's the name of this organelle? Anybody can tell me the name of that organelle? It's not on the biology syllabus. It is on the HSB syllabus. No, it's not the ribosome, but it would contain the ribosome. It is on that we're going to find the ribosome. That is called the endoplasmic reticulum, right? And there are two types. You have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And the dots that you have on it, those would have been the ribosomes, and that's for HSB. All right, so I was just pulling you there a little bit just to make sure that you understand. And having damaged you so much with the questions we asked before, I decided I'm going to give you something a little simpler. So we would have answered all this question here when we actually, you got 6 marks there for identifying the parts of a cell. So we want to move on down, and now it comes a little tricky. Now you're going to differentiate between plant cells and animal cells by identifying 2 differences. Explain how each difference is important to the plant existence. All right, so we're going to be answering this question in the chat now. I see that persons are waking up. Earlier, I was a person answering the questions, but now I have persons who are assisting me, and I'm most grateful for that. I really don't like to come to teach and I'm telling you everything, right? I want to hear from you. I want to correct you. I want to get rid of misconception. I don't want to come here and just drain things from my head to yours. I want to know what you know, either to remove something or to build on something. All right? Because I'm sure that you come with something. Wrong or right, you have something. All right, so is cell a topic on the syllabus, on the broad topics? One thing you're going to understand that those broad topics in some cases, they are pretty vague. And you have to, I think, cell, if you look at movement, there's no way you're going to talk about transport and you're not talk about cells, because transport has the blood cells, so you must talk about cell. So while cell might not be explicitly named, there are some things, though they are not well named on the broad topics or in the broad topics, we still have to cover them. There are some that we don't need to cover, like respiration, though it will be on your paper number 1. All right. So the plant has a cell wall, however, animal cell does not. All right. So we've seen a number of stuff here. Let me just show them on screen. So we have plant cell contains cell wall and chloroplast. But what happened to animal cell? That there's no comparison there. So you have to tell us about the animal cell. You can't just tell us about one. All right, so let me see what's happening here. The plant has cell wall, however, animal cell does not to support the plant structure. So you are pretty much answering a whole lot of things here. And then let us move on down here. We have another one. The animal cell, there is no cell wall, while in plant cell, there is, of course, cell wall, which protects the plant cell and gives it shape. All right, so I think how many we're supposed to do? 2. Just let me look at that. It's a differentiate between, and it said 2 differences. So I'm seeing 1. In some cases, some persons are talking about the cell wall. In some cases, persons are talking about the chloroplast. Those are 2 nice ones. We could talk about fixed shape. We could talk about this where the nucleus is. We could talk about, you know, the best things. But I think the best thing that I would go for if I were answering the question like you is, of course, to go for the chloroplast and, of course, go for the cell wall. Those really much stick out like a sore tongue. The explanation is very easy when we are using those. All right, so just make sure that we do that. We would have said what the differences are, and we are going to explain how each difference is important to the plant. So while somebody said there that the cell wall helps the cell to be rigid and give it shape, we have to also be reminded that the chlorophyll is very thick. What is it? The chloroplast is very important for the process of photosynthesis, as it would have contained that chlorophyll to trap the sunlight. So we have to make sure that when we are answering these questions, we're answering all parts of the question and not just the easiest part if we are going to be securing full marks, because the whole intent is that we want to come out of that exam with the best possible marks. So we want to do that, we'll have to answer the questions appropriately. Now here we want to state the expected observation. Look at this one. We want to state the expected observation in each of the cell type. Now plant and animal cell was placed in distilled water for 15 minutes. All right, so distilled water is that water that you would have used in your labs in most cases. So just one reason for each observation. So when we put the plant cell in the water, what observation there will be and what's the reason for what you would have observed. All right, so I am now looking in the chat to see how you're going to treat with this particular question in order to score full marks. Please be reminded you are watching CSEC Biology TCP or CSEC Biology the cover page. I am Mr. Wilson. We're streaming from Kingston, Jamaica, and the sole intent is to help you get that A for biology and HSB. Now, we are pretty much on air. Well, we are going into holiday now, so I'm not too sure if we're going to be on air, but you are seeing streaming online. We have TCP Academy. That is our paid platform. So wherever you are in the Caribbean, we have a package just for you, and you want to, as much as possible, give us some love. Now, if you're joining for the first time, welcome to the show. Welcome to Mr. Wilson's platform. I'm really, really happy that you are here. Please be reminded, however, to subscribe if you like what you see here. And of course, click that notification bell and select all so you'll be notified as soon as there is a new publication, or if I have to send out a post, which is not a video, just to inform you of something, it comes right to you. If I were supposed to get some breaking news out of CXC or breaking news, read the content, when I send it, it just pop right up to you if you click the notification bell and select all. When you watch a video, if you want to add it to your playlist, it would be wise if you click that up arrow, that up thumbs up, and then it would have been added to your playlist. If you have not yet subscribed, it is time now to subscribe. I will want you to pass this information on to students in lower school, grade 10, that they will come on early, not 2 or 3 days before the exam, where we can assist them in preparing for the exam, be it on our paid platform or on our YouTube channel. So let's look at what is happening here. I don't like when students write and use it. All right, I prefer if you say the plant cell would become turgid. All right, so that is an observation that you would have had. So here I'm seeing one person saying the plant cell will become turgid. I'm sharing on screen. And here I'm seeing another person saying it will become turgid, but will not burst. This is very, very important because of its extreme armor. And that extreme armor, extra armor there is, of course, going to be the cell wall. This is a wonderful answer, I think. And then we're going to move on over here. Plant turgid structure. This is due to the process of osmosis. What I'm looking for, yes, osmosis is happening. I'm not sure if it's that you're answering to the same question or the question that comes below, but I think I'm not finished fleshing out this particular question. There are some things I'd love to talk about before we move on to the final process there, which is, of course, osmosis, and you are correct. But we are not at the end there yet. We haven't looked at what will happen to the cell, the animal cell having been placed in the water. So we want to look at it here. If we were supposed to place a plant cell in water, what would have happened is that, of course, the cell would become turgid as a result of water moving into the cell. Now, we are saying the reason for this is because of a concentration gradient that is created as a result of a cell being in what we refer to as a hypotonic solution. So with the hypotonic solution, what is happening here, and we're looking at osmosis here, what is happening here is that with a hypotonic solution, the outside, the water, the content on the outside of the cell is going to be less dilute than the content inside of the cell. So what you're going to have happening is that water is going to move into the cell. Now, based on the plant cell with its rigid cell wall, the cell will not rupture. But let us look at what will happen to the animal cell. Will the animal cell be pretty much the same as would have been with the plant cell? So let us look at this one. What will happen here? What will be your observation for the animal cell? What's your observation there for the animal cell? All right, so we would have placed this animal cell in distilled water for 15 minutes. What is likely to happen? And I do have a video on my channel that cover all this bit of content. So when we place the plant cell in the water as we were doing osmosis lab with the potato strip, you would have observed that it became turgid. So I'm seeing Philip coming out of the block here, and Philip is saying that sir, it will burst. All right. Interesting. Not too sure why it will burst. So you want to give us the reason why you think it will burst. And the plant cell did not burst. So share the answer there with us. I'm going at your speed. I'm pretty much listening and working with you. Because like I have said, I really don't want to give you the answer unless you're struggling. I really want to hear from you so that we can correct some of the ills and strengthen some of the strengths. All right, great. So you are watching Mr. Wilson live. We're doing our marathon. We are working the 2020 January biology paper 2. We would have gone through 2019 already. Last night we went through 2017. And I think we have the 2021 and 2018 paper left to go through. Now, I'm going to try to see if I can upload these papers for those persons who were not following our persons who missed it. You will find the paper and the answer hopefully today on the channel, hopefully. All right, so I'm seeing no further answer. So I will just delve right into the animal cell. Now, the animal cell being placed in the water is pretty much a hypotonic environment, just the same. Now, the animal cell is a little more, okay, sir, when the water enters the cell via osmosis, because the cell membrane isn't, okay, let us see this. Sir, when the water enters the cell via osmosis, because the cell membrane isn't strong because of a water pressure, the cell will eventually burst. All right, so the cell will eventually rupture indeed as water enter the cell. So that is really true. So water is going to go into the cell because of that concentration gradient, which brings me to the point. I have never ever taught osmosis and not made mention of a freshwater fish versus a saltwater fish and IV. So when you go to the hospital and you are given what we refer to as drip or the intravenous fluid, it is not water as in distilled water. It is more salt water. Now they have to ensure that the content that they are putting in your body, it is pretty much in an isotonic state. Isotonic state. So in the isotonic state, what happens is that the concentration of that drip is pretty much a concentration of the body. So you won't have this water flowing into your cells to burst the cell and kill you. Now the same is true if you take a saltwater fish out of the sea and place it in the river. It will die because the water from the river will move in as there's a big concentration gradient, burst the cell and cause the fish to die. Except I think for the salmon is able to pass its urine, so this wouldn't affect the salmon. The salmon can go from sea to fresh water. Now the same is true. You can't take a freshwater fish. You can't take a saltwater fish from the saltwater and place it in the fresh water because now it is now exposed to a hypertonic solution where of course the water will now move out of the fish into the salt water and so too the fish will die. So remember those three words: hypotonic, hypertonic, isotonic. Those are all nice words in osmosis, and you might have heard about plasmolyzed, flaccid, and of course turgid. All right, great. But I think you did well with that just now. And for those persons who didn't know all those stuff I said just now, make sure that you jot them down somewhere. And the final question here is to name the process which is responsible for the observation in BI. So what process is responsible for the observation in BI? And the one we just answered just now, that's BI. What process is responsible for that? All right, let me see. All right, so girl is out of the block pretty early, and girl is saying that it is osmosis. There's somebody else out of the block. Let me see what's happening there. All right, Carlene Frankson is also saying osmosis. Both persons are correct, and I'm seeing Tori. Tori is also saying osmosis. All those are correct, and I'm seeing somebody else coming in with osmosis. Ruth is also saying osmosis. Wonderful. Now, the next question we are going to be looking at, the next question on the paper is going to be the I, but the I you told me last night is not on the paper. And that worry me somewhat because I've been seeing so much about the I that I am wondering what question will come there. In my mind, it is going to be something on transpiration or it is going to be something on circulation. That is my view, that this question might be replaced with a transport question. Be transport from plant or transport from animal. One of the two. Because being that the I is going away, something must take the space. Not too sure, but my assumption is that it's going to be transport. Now, interesting, we're at question number 4. Now, it's section B, and you're supposed to answer all the questions. You're supposed to write your answer in the space provided in this booklet. Now, figure 3 is a simplified diagram of the carbon cycle. Now, we have to get pretty used to the carbon cycle. I don't think I really like the nitrogen cycle. I think it's a little complex. I don't know. I just really don't fancy that cycle. But I really like the carbon cycle because the carbon cycle pretty much has 4 processes. It's going to be photosynthesis, respiration, fossilization, and combustion. Only 4 processes we have in the carbon cycle. So what I want you to do, I want you to identify, look at these, I'm not too sure, I'm not reading well now. These, we want to identify these processes. All right, so let us just go down. So we're supposed to identify the parts labeled A, B, C, D. So there they are. Please identify them in the chat for me, what is happening in each area. Tori, are you doing the alternative paper? The nitrogen cycle is not, I don't think it's on the bio syllabus. It's a biogeochemical cycle, but I think it's not there. I think it's on the HSB syllabus. All right, I don't know what's wrong, but I'm not breathing so well. All right, so let me look at some of the answers that are coming in. I'm going to be posting them on screen. All right, somebody said that D is combustion. Somebody said that C, decomposition. I didn't see that coming. I didn't see C coming, but let me see what other persons are saying. B is, of course, photosynthesis. Oh, so Cheryl, you are doing the alternative paper. So I keep saying, Tori, if you are doing the alternative paper, you need to look at my videos over there for the labs and all of these question number 1 that we have been doing, Tori. You need to know them because all the question number 1, they are labs. All right. So let me see this one. This is a combined answer. Let me see. So A here is going to be respiration because the tree is giving off things to the cloud. B is going to be photosynthesis. And C is going to be decomposition. Might I say I have something different for C? And D is going to be combustion. But let me go down. I am not going to act as if I am the keeper of knowledge, but I am going to defend my point, and you will have to defend yours. All right. So we all agree on A being respiration. Tell me and get ready to fight. I'm going to fight you. So get ready to fight. I am going to fight you. All right. So A, respiration, we agree on that. B, photosynthesis, we agree on that. D, combustion, we agree on that. But C, we can't agree on that. Now, you will have to defend your point because I frankly don't think that that's the answer. But like I say, I am always willing to change. So let me just zoom up and let us have a fight over this now. Can you see the screen? I should have made it, I'm going to make it much bigger. All right, just give me 1 second. What do I need to do? All right, so I'm going to carry it here. And then I'm going to carry it. Don't do that to me. Now, look at what is there. We are concerned about C. Tell me if your position is the same. Don't be afraid of me, you know. If your position is the same, your position is the same. And if you think you should change, change. So now it's big on the screen. I want you to tell me what is your position. Do you maintain the position? Do we have to fight over it? What is your position? It is not, Philip. I am not going with that answer. I don't think that that is the answer. All right. What is the answer, sir? Why is it that you think I should tell you everything? I want you to tell me everything. So tell me some, just make some, okay, great. So Lee, you think that it is fossilization. Can you support that? Yeah, it looks like decomposition. Definitely it does. But let us look at some stuff. I'm going to go over there and I'm going to be on Lee's side and I'm going to show you some things why I don't think it is decomposition. Now, do you realize that, let me just change the color of my ink. I am going to use blue. All right, do you observe that this is the soil? This is the layer of the soil. Look at this, layer of the soil. Do you take any clue from this being fossil fuel? Now, if you look at this here, these look like the skeletal remains because there you are seeing the rib cage, right? And that is why I zoomed just now. Look at what the cattle, the remains of a cattle versus the remain of a pig, So I am seeing here, in my opinion, that this is pretty much fossil. Now, I'm not too sure what this arrow is saying. Or it is saying that when it dies, it gets here, and then it also will be converted into fossil fuel. So I personally think that it is fossilization, but of course, if you can, I think, in my opinion, a cattle and a pig dying, they would have decayed on the surface of the earth. But I think that the fossilization is going to be underground with temperature and pressure and all of that. So I strongly think that it is, of course, fossilization and not decomposition. If you are not in agreement, that's fine. But that is what I think. All right. Can we move on? We have a fix. We still have anybody want to go other way or otherwise? All right, so there's a lot of things, there's a lot of clue in the question. This is another clue. Look at the fish here. Look at the level of the soil. So the level of the soil here, you know, and the fish is way down here. And look here, we have some reef. I remember that the reef must be on the sea floor, right? So this is pretty much, if you look at the water column, the reef here is like a benthic organism. And the reef here dies and the reef enter this thing as well, right? Enter this fossil fuel thing and we're going to get oil and coal. Remember, oil and coal are fossil fuels, so fossilization must take place here in order for us to have fossil fuel. So pretty much that's my line of argument. I'm not too sure how other person would have argued it. All right, recently there has been an increase in human activity which resulted in the increase of atmospheric carbon dioxide. And I think I really want to work on this question with you. So just 3 human activities that could have contributed to the increased level of carbon dioxide in the ecosystem. And for each activity, state one way by which the increase in carbon level, one way by which it increases carbon level in the ecosystem. I hope I read that right. If I didn't, I will read it again. All right. So let me just come back over with you and we'll talk about that. So I'm going to read it again. Now, recently there has been an increase in human activity which resulted in the increase of atmospheric carbon dioxide. Suggest 3 human activity that could have contributed to the increased level of carbon in the ecosystem. And for each activity, state one way by which it increases carbon level in the ecosystem. Now, I am going to love your answers here. I understand. Sir, have you all done anything with variation? Well, we were doing some variation videos earlier. We did a paper last night on variation, and this paper has some variation at the end of it. We're looking at the 2020 January bio paper 2. All right, so let me see what you are saying now. And remember that this is my baby ground. This is environmental science. All right, so let me see. Okay, burning fossil fuel, that's a good place to start. So let me go with Philip. I'm going to start with, oh, wow, where's Philip? Philip up the way. All right, great. So Philip is on screen. So burning fossil fuel when you burn fossil fuel, carbon dioxide is given off, and that, of course, increases atmospheric uh, carbon dioxide. Now, the AC unit, what about the AC unit? I don't want to help you. So the AC unit would pretty much be in keeping with this one here, the CFC, chlorofluorocarbons, and of course burning of fossil fuel. But it said three ways, and I'm seeing two so far. So let me look here. Oh, beautiful. It's coming up nicely, man. Deforestation, by decreasing the amount of tree, there is less use of carbon dioxide for photosynthesis. So remember that trees serve as what we refer to as carbon sink. So what carbon sink means is that it pretty much store the carbon there. And if it is a sink, it is a source, but depending on where it is. But of course, trees serve as carbon sinks. So when we deforestation, because of deforestation, we are pretty much adding indirectly carbon to the atmosphere, as we are not able to, of course, remove it by the plant, because we actually remove the plant. All right, let me look at this other one. It says here that deforestation, due to this, there would be less plants for photosynthesis to occur. All right. So photosynthesis uses carbon dioxide, which would have removed carbon dioxide from the air. So we understand that that is also plausible. So far we are looking at about four things. We are saying that because we burn fossil fuel, we are adding carbon dioxide to the air. Because of deforestation, we are adding carbon dioxide to the air. And somebody said chlorofluorocarbon, that also. But when you get to the exam, you really don't have to say fossil fuel. You could speak about burning coal, burning oil, burning gas. It could be said that way as well. But of course, the CFC is a nice little additive here. Now, this one I like here. Again, it says, it should be on the screen soon. I don't know if I have a lot of things opening on the screen. I'm seeing some things in the background. All right, great. So this one here says deforestation remove trees and cause the concentration of carbon dioxide in the atmosphere to increase. This is because trees, through the process of photosynthesis, help to remove carbon dioxide. Great. So a lot of persons are on the right path. I wasn't expecting this one any at all. Let us look at this one. Is it plausible? Overpopulation is being argued here. Because of overpopulation, what is happening is that we have more carbon dioxide being expelled from organisms, so that is assisting. I didn't see that, and we could also look at overpopulation with the consumption of products, and then as we try to get rid of them, that is also adding. But that is sort of an indirect phase to what we're looking at. The other answers are a little more spot on, but I think I like that attempt as well. Transportation would also be involved in public transportation would also be involved, whichever transportation, in the burning of fossil fuel, and that could also increase the amount of carbon dioxide in the air. Now, sir, can you explain chlorofluorocarbon? All right, chlorofluorocarbon. I'm not a chem spec, but it is something that is used with refrigeration process, cooling. And it is considered to be one of those greenhouse gases. But the fact that it has carbon, because it's a chlorofluorocarbon, so I think it's something from root 7 of a periodic table, then that also would add to a carbon. But the popular answer here is always going to be the burning of a various type of fossil fuel, whichever way you burn it, and of course deforestation, whichever way you carry out deforestation, right? And remember, I think it said that you are supposed to explain. I don't remember if it said that. If it said that, you really want to ensure that you are answering the question correctly and you are explaining. So it says for each, just let me read that for you. So having suggest 3 human activity, then you are supposed to say for each activity state one way by which it increases carbon level in the atmosphere. A lot of persons went out and they tried answering the question to ensure that they got this in. But remember, when you go there, you have to ensure that you're answering the question to obtain full marks. I really don't want to answer just a part of the question coming out of the exam saying that you have scored 100%, only to find that you just got 1 out of 4. Now, suggest 3 practices that can be adopted to reduce the impact of human activity on the carbon cycle. This is going to be interesting 3, and there are tons out there. So I want you to give your 3, and having given your 3, I will just add in some more and explain them so that they become a part of your vocabulary, they become a part of your learning. So what we want to know here, we want to suggest 3 practices that can be adopted to reduce the impact of human activities on the carbon cycle. So somebody is saying using electric car. All right, so somebody say use electric car. I think I'm going to have to talk about that, using electric car. So when we use electric car, what is the real source of electricity? So this is another nice word. I've seen some nice word coming up. Could we explain reforestation? What does it mean? What does reforestation means? Okay, we could grow more trees. So reforestation, I'm seeing grow more tree. We could increase the practice of reforestation. We could recycle, of course, use biodegradable material instead of burning. We could do some carpooling. But what I realize is that I'm getting a lot of low-level answer, and I'm seeing just one high-level answer so far. Now, we have to take you away from primary school and give you new content. I'm also seeing replanting trees, all right? And you're not using the term that is on the syllabus. Replanting trees, you're not going to see that on the exam paper. You must use the correct term. Reforestation is growing of plants. That is vague and really don't mean much. It suggests to me, read, that you don't know what it means, so we'll have to assist you here. Now, reforestation is the planting of trees back into the environment. I hear you say that, but I think that is still over your head, Philip. So we have to really get that into our head, what it really means. Now, planting plants in an area where there have been removed. Now, I think that this one is probably the better answer for reforestation. So if we are going to, if it's reforestation, it's the same thing as replanting trees, but on your exam paper, it's going to be reforestation. Once it's reforestation, it means that you are putting back plants where they were before, which brings me to the other point that is in the chat now, replanting trees, afforestation. What does afforestation mean? And these words are always on your multiple choice paper. And when they get to your multiple choice paper, because you are so accustomed to replanting trees, when you see afforestation and reforestation, you are not too sure what to do. And when you see conservation, that is also giving you a fight. So we want to know what it means, afforestation. What's afforestation? Develop education program, replace trees, practice organic agriculture. That sounds pretty nice, Lee. It sounds like you guys are all environmental science students based on the answers you are giving here. So I want to know what is afforestation. I'll quickly show what Lee said on screen, and then I'm going to be showing you what Charles is saying. Let me read that for you. It says here that we are supposed to reduce the use of transportation that requires fuel for combustion when, sorry, suitable walking instead of driving. All right, so I'm seeing a lot of things. I'm seeing that you guys know a lot about your agriculture, reforestation, and all of those stuff. Okay, finally, I've seen somebody coming up with some technology in the mix now. So somebody is saying here, Francis is saying that using solar energy instead of fossil fuel, that is a good move as well. I think I like that. So putting some technology in the mix rather than just planting trees, let us just move away from fossil fuel and go to renewable energy where we can use wind, water, all those other sources of energy. We can harness them. Good morning. Are you going to be live on Sunday? Yes, I'm going to be live on Sunday because we have two other papers, I think, to cover. So we're going to be having two shows on Sunday. We're going to be having a HSB straight paper because I don't want my HSB fans to kill me. They have been here with me for over a year, and I'm sure that they are mad at me now because they have been here all along. And I don't see one of them in the chat now, so it means that they vexed me. All right, TCP. So reuse, recycle products. Example, plastic. Uh, so great. So I have seen a lot of wonderful examples, but I think I wanted to look at something that keep eluding me here. And this is what I want to look at. It is on screen now. Now, afforestation is planting trees where there aren't any trees. So when we talk about afforestation, we are planting trees where there were not, where there are no trees or no tree was there before. That is afforestation. You are introducing the plant. With reforestation, there was a plant there before, or there was a forest there before. So that's the difference between re and a. A, there was no plant there before. There was no forest there before. Re, there was a forest there before, but they cut it down for yam stick, they cut it down to burn coal, and we have to now replant it. All right. HSB students are going to be having live session, hopefully from Sunday, Monday night, Tuesday, Wednesday morning. So I'm going to be having live session with them all that time. I don't know what I'm going to tell my family because I've been up from 2 o'clock trying to make sure that I would have been here for you. I was here from 4:30 doing this, running some videos because I have a very short day. So I try to accommodate you when I can so that I can get the rest of the day to do some stuff. But person didn't come on until late. I really didn't like that, but I understand that persons were asleep. Not everybody's like me, but I try my best to fit you in. I understand what it is like, more so with COVID. Some persons were not in touch with school and all of that. So I am doing my best. Though I don't know anyone here, I am still doing my best to see if I can help you. Someday you'll help somebody I know or don't know. All right, so we're seeing a lot of stuff. So I like what I'm seeing, and I have slowed down the class a while ago just for you to just tell me what you know so that I can correct. So for me, my answers would have been the first thing we want to go to renewable energy. Now, the renewable sources of energy, I have to give you information now to go to sixth form. I cannot send it to sixth form empty, and that is why we looked at reforestation, afforestation. So when we talk about renewable sources of energy, we are talking about things that when they are used, they are not uh, exhausted. So we could talk about using solar energy, which is big in the Caribbean. And we can harness solar energy through photovoltaics. That is what we call the solar panels. Or we could harness the thermal energy, the water heater. Then we have wind energy, which is harnessed via the windmill. That's an alternative source of energy. Those could have been used instead of fossil fuel. Then we have wave energy that come from the sea, wave coming in and out. And then we have tide energy that come from the sea, the tide going up and down. Then we have hydroelectricity that come from rivers and dam with the water flowing down. If you are doing physics, you would have learned about potential energy. Now, we also have geothermal energy that is coming from hot spring. All those are alternative sources of energy. And then there are some new technology like using water to make electricity in what we call the hydrogen fuel cell. Now, that might be a little too much at CXC, but that is something that we do at environmental science as an alternative source of energy. So to deal with the problem, to arrest the problem of carbon dioxide emission, we could turn to alternative sources of energy. Now, nobody said this, but we could have used what we refer to as the carrot stick approach. Now, the carrot stick approach is that if you are, and I'm going to give you an example in Jamaica, between Jamaica and China. Now, the carrot stick approach is that you provide incentive when persons do something good, and there's a penalty when you do something bad. So you could pay persons to plant tree. For companies that have like carbon scrub on their chimney, you could give them a tax break or give them some incentive. Now, if they breach and they are putting out more carbon than they should, then you could use the law to go against them. That is going to be the stick. So the carrot stick approach could have been used, and I would have explained that to you. We spoke about reforestation, we spoke about afforestation. Now we spoke about planting tree. Now there's also what we call catalytic converters. Catalytic converters are now on most motor vehicle, and what they do is to capture that suspended solid carbon particle and of course keep it and empty it somewhere. I don't even know where, but it happens. So you could use catalytic converters on motor vehicles. Always remember that you might see what is it that you can use on a motor vehicle. I have seen it on the HSB syllabus. I have seen it on the biology syllabus. So always remember about catalytic converters on motor vehicle. As it were, for smokestack chimneys, you can use carbon scrub to remove the carbon. I wanted to say something else and it slipped me, but I'm going to give you before I go. I'm giving you everything on this thing. All right, so another thing that we could use is what we call carbon capture, which would have been like the catalytic converter and the carbon scrub, where the fossil fuel that is burned, now artificially the carbon is captured and it is buried underground, like in crevices and places where we mine from, we bury it and all of that. All right, so that could have been done. I said I was going to tell you something about Jamaica and China. Now, in Jamaica, because China is producing so much, they have to do a greening project. So there is an agreement in Jamaica between China and Jamaica with Wigton Wind Farm. I don't know if that has changed. And Wigton Wind Farm is our wind electricity farm in Jamaica. I think I'm not saying something right, but just get it. The wind. So we have electricity produced by wind over there in Manchester. When this happens, what happens is that depending on the amount of megawatt of electricity is generated, you get carbon credit. Now the carbon credit is sold to China. So listen to the hypocrisy. So while China is greening in the Caribbean, they are still polluting in China. But that is another thing when we spoke about the carrot stick approach. They are doing some pollution. They are pretty much manufacturing for the world, but they are also using their good citizenship to ensure that they are greening projects across the world. I do hope that you got those. I said a whole lot just now. Some of it is a little bit above you, but because I know that most of you will be going to sixth form, I gave you a little bit of information that when you get to sixth form, you won't be all blown away. Now, human activities can lead to an increase in greenhouse gases. Now, you are supposed to name 2 greenhouse gases other than carbon dioxide. Tell me in the chat your greenhouse gases other than carbon dioxide. Remember that carbon dioxide is the 4th most abundant gas in the atmosphere. So 2 greenhouse gases other than carbon dioxide. Yes, and remember that methane is, of course, produced naturally. Each time that a cattle would belch, methane, and as things decay, methane is also produced. So I'm seeing methane and I'm seeing now nitrogen. So we talk about nitrous oxides. Those are also greenhouse gases. And we have a chlorofluorocarbon, that's also greenhouse gas. And surprisingly but true, water vapor is also a greenhouse gas. I think you all did well there. Good morning and welcome to the show. You are watching CSET Biology TCP with Mr. Wilson, and we're streaming from Kingston, Jamaica. Now here we go. Question number 5 with just 1 to go. Complete the following table to compare the disease hypertension and yellow fever for each characteristics. Now, let me just look at you and talk to you something here. I want you to pay attention to this table. I remember when I taught on our live session about mosquito as a vector, and we're looking at the many diseases. The popular diseases on your syllabus, is malaria and dengue. Now, when I check the biology book, I can't remember seeing yellow fever in the biology book. But the HSB text, it has yellow fever. So most of you will be going to the exam and you are going to be armed with information about dengue and malaria, but not about yellow fever and not about this other one that is spread by the colics mosquito. Just let me just remind myself, because from time to time I'm getting so old I forget things. But there's another disease there being spread by mosquito, the same Aedes, I think, is spreading a disease out there that it's not yellow fever. Let me tell you what it is. I'm getting old. I think I'll have to borrow one of your brain one of these days. It's called filariasis. So filariasis, and this is pretty much a worm that is spread. You want to make sure that you know these things. What I want to really point out here is the whole idea that from time to time persons pop on and there is this bio or HSB. Oh, only the plant aspect is biology. The rest of it is HSB. So here, what we're looking at, this is on both syllabus, both biology and HSB. You cannot go to a biology exam or HSB exam and not know about diabetes and hypertension. They are on every biology paper. Diabetes and hypertension. So here we have yellow fever, and a lot of persons would have been bowled by yellow fever because you were all prepared in most cases for malaria and dengue. Now, what we're required to do here, we are supposed to compare. So we are supposed to tell now hypertension what category of disease this is. All right, so some hypertension, persons are out of the block early. So hypertension is a degenerative disease I'm seeing on screen. Anybody else with anything different? Hypertension is what type of disease, and yellow fever, what category of disease these would have fallen in. All right, so we have seen degenerative disease. Let me see what's happening here. All right, so it can be degenerative. Please do not write your answer like this. While I understand the other students who are watching might not. So of course, hypertension is a degenerative disease or a physiological disease. If you ask me, you really don't have to get old to have high blood pressure. There are a number of ways that you could have that, but it is categorized as both degenerative and physiological, or they are used interchangeably. All right, so we are seeing physiological there from Philip. So we move on down now to, we have seen on screen, not something new coming up on screen. Hypertension is physiological disease, while yellow fever is a pathogenic disease. I like the way Stuart would have answered that question, and we need nothing less when you answer the question, so we know exactly what you're talking about and there's no misconception. Now we're moving on to another answer, and we're sharing the answers on screen in most cases. Hypertension is a physiological disease, while yellow fever is a pathogenic disease. And I think for most cases here, everybody would have gotten that correct. But look at this one down here. If you were supposed to see infectious disease, it's not on screen yet. All right, infectious disease, yellow fever. Infectious disease, they are usually pathogenic disease, but usually the category is pathogenic, but I do understand that infectious is the same thing as pathogenic disease. So yes, a lot of persons are doing well here. Congratulations. Let's move on with the content. And you got 1 mark for that. Now, what is the disease caused by? So we want to know now, what is hypertension? What causes hypertension? What causes hypertension? And what would be the cause for yellow fever? Tell me in the chat the cause of hypertension and the cause of yellow fever. Oh, that's a mosquito. All right, great. So the cause of hypertension and the cause of yellow fever. Remember, please do not go to the exam not knowing about diabetes and hypertension. That is like a giveaway. You're sure that that is going to be on the exam, on both exams. All right, so there's somebody out of the block saying that high salt intake might be responsible for diabetes, hypertension. Somebody is saying hypertension. Yes, it is high blood pressure, but what really causes it? Now, the truth is that the disease is caused, once it's a physiological disease or a degenerative disease, it is pretty much caused by malfunction in body parts. And malfunction might mean that something is not, it pretty much it means that something is not working the way it should. So even though we are going to say that it could be as a result of high salt intake, it could be as a result of poor eating habit, it could be as a result of sedentary lifestyle. Now, all of these are going to lead to malfunctioning organ. All of these will, of course, lead to malfunctioning organ. All right, great. So we have a clear understanding there that once it's a physiological disease, it is caused by malfunctioning organ. And here we could give example with salt, how the salt would cause it to malfunction. We could also give how the, for example, if you're taking in too much fatty food, it's going to cause the lumen in the blood vessel to become very small. When it becomes very small, then you understand that the blood is naturally going to be under high pressure. I want to tell you this before I move on. Now that I just remember here before we move on over to the cause of yellow fever. A lot of persons, you are supposed to, from you reach age 18, you should begin getting regular checkup of your blood pressure. Blood pressure, high blood pressure tend to go unnoticed, undiagnosed for years, right? Now, when you do your blood pressure tests, Amoy is here. Good morning if you are here, Amoy. I realize you're not talking to me. You should do the blood pressure test on both arms, the right arm and the left arm. Now, your blood pressure is measured in something under mercury. Oh my, it just slipped me just now. However, there are two things that are being looked for when we're doing your blood pressure test. We're looking for the systolic reading and the diastolic reading, which the upper one is going to tell you the blood pressure when the heart is beating. And the lower reading is going to tell you the pressure in the blood vessels after the beat. So when you do your blood pressure reading, you'll realize that it is giving you two numbers. One, the first number, is telling you the pressure when the heart beats. Boom! And the other is telling you the reading after the heart stop beating. All right? High blood pressure can be caused by overweight. Yes, true. Now, let us look at the reason now, the cause of yellow fever. Tell me the cause of yellow fever there in the chat. I think I'm liking it, only that you guys are late and you're all supposed to go into the naughty corner because you were late for class, having agreed that we're going to have class 5 o'clock this morning. We started class late and some person missed parts of a lesson, which is not nice, knowing that your exam is coming up in a few days. Yes, we are doing the 2020 January paper. Great. We are doing the 2020 January biology paper 2. All right, great. So let us continue. What causes yellow fever? You are supposed to tell us that. So we are at question 5 with question 6 to go. And the last question on this paper, I don't know if you realize, on all the papers I have worked so far, there is a genetics question. So we have a genetic thing here that we all will have to work out. And I'm going to just give you a little bit of heads up on genetics as to how the question could change. What is it that they could ask on genetics? But I am almost certain, based on the trajectory, that genetics might come because genetics falls under variation. Yellow fever is caused by a pathogen in the case of mosquito. You're not going to take any shortcut on me here. Oh, no. Philip, you can't take a shortcut on Mr. Wilson because I could name out a long list of pathogens. Are you suggesting that yellow fever is caused by a protozoa? Is it caused by a bacteria? Is it caused by a virus? Is it caused by worm? What is it caused by? So don't take that shortcut on me. Be a little more specific, please. We know that you are hiding something when you are not shooting the way we want you to. All right. So now you're laughing. So you should go to the naughty corner because you know what you have done. You know what you have done. All right. So yellow fever is caused by the Aedes mosquito, which, all right, cool. Let us look at this answer. I hope I put the right thing on the screen. All right, great. So yellow fever is caused by the Aedes aegypti mosquito. Now, yellow fever is not caused by the Aedes aegypti mosquito. Yellow fever is caused by the virus that is transmitted by the Aedes aegypti mosquito. Now, if you are going to say that it is the Aedes aegypti mosquito that causes yellow fever, then pretty much we are going to have yellow fever each time we are stung by one of those mosquitoes. I remember that mosquito would, of course, transmit other type of disease. So, of course, the mosquito is just the vector or the bus that is carrying you from one bus stop to another. Now, the virus is what really causes the yellow fever. All right, great. So the pathogen that we're talking about here is the virus. I see you guys jumping around the place, which means that you don't know what are pathogens. You just know that it's a disease causing agent, but you can't tell me that pathogen will refer to like bacteria, virus, fungi. There are 5 of them. Protozoa, and what is the 5th one? All right, great. So the next thing that we're supposed to look at, let me scroll down. I think you've been nice so far. Just let me scroll down here. One sign symptom of disease. So I want to know a sign or a symptom. And before we answer to sign or symptom, you will have to distinguish for me between sign and symptom. What makes the difference between sign or symptom? What's the difference there? Yes, organisms that cause a disease, they are pathogen. I would say that there are 5 types, 5 groups of these organisms, and I gave you 4, and I asked if, in a matter of fact, I gave you all, and then I gave you 4 and asked you to give me 5. I gave you all 5 at one point. All right, great. So now I want to find out the difference between sign versus symptom. You know, like all the while when we're writing up that lab, we say apparatus material, but we really don't know the difference. And we always talk about signs and symptoms, but we really don't know the difference. Now we want to find out what are the differences or what difference there is between sign and symptom, or I want you to define sign and define symptom, and then we are going to come back to this particular topic, particular question. All right, so let me put this on screen and we can all share in the response here. Don't understand. Oh, that's not the one. This is the one. Hey, why are you doing that to me? I think I'm doing something. It's slow and I keep clicking. All right, a sign is something that can be seen physically, and a symptom deals with internal health. For example, having a fever. Can I tell you something? This is one of the accurate, inaccurate answer. I don't know if I make any sense here. I need to put that in some literature. It is an accurate, inaccurate answer. And I'm going to explain what makes the accurate, inaccurate answer. But before I do that, just let me look at the others. Let me see Stuart. I'm going to come back to your answer. And if I don't, please ask me to. What makes it accurate, inaccurate? An individual, I would probably not feel the sign, but may see may see it, while they might feel the symptom but can't. Oh my. I'm going to go to my answer before. I don't remember who gave the answer, but I am going to go back to that. Who was it again that gave that accurate, inaccurate answer? Let me look at it. Sign will be, where are the students that I teach for 1 year? Where are they? I haven't seen one of them. What have I done wrong? Sign will be the physical sign that tell you that a person has a specific disease. Guys, I am so happy I asked you this question because you'd go to the exam not knowing the difference. You have an idea, but you can't say for sure what it is. A sign is what can be observed by another person, and symptom is what the person, this is a Jamaican speaking, symptom is what the person feel, which cannot be observed physically. Anyhow, I'm going to stop harassing you now, and I'm going to assist you. That reference that I made to the answer that I read first, this is my explanation. Somebody remind me of the name so I can find it, so I can just dissect it and treat with it. Okay, I find it. It's right here. Let me put this on screen and let us correct some misconception here. All right, it says that a sign is something that can be seen physically, and a symptom deals with internal health. For example, a fever. Let me correct this misconception quickly. And if your teacher has something wrong, different, just ask them to send me a mail and we can have a discourse on this. When I taught disease earlier this year to my online class, I had a CXC question and I'd asked them. At that point, I learned that the thing that you used to put on your tongue, the foot stick that the doctor put on your tongue, Amai told me that it is called a tongue dispenser. Amai, you see that I'm learning and not as dunce as you think. I remember that which you taught me. It is called a tongue dispenser. I am going to Amai's tongue dispenser because if I look at the definition here, it says internal health. At that point, students could not appreciate the fact that a sore throat is a sign. They were saying that a sore throat is a symptom because you can't see the sore throat. So I'd ask them, when you go to the doctor and the doctor asks you to open your mouth, stretch your tongue out, they put this tongue dispenser, I can't say foot stick, I don't want Amoy to hit me. Put this tongue dispenser on your tongue and see that sore throat in the back of your mouth. Are you going to tell me that's a symptom? No, that is a sign. So it is true that sign refers to what you can see. But hold on. Sign doesn't only refer to what you can see. It is true that sign refers to that which an external source can detect. So if you are having a fever, I could detect that you are having a fever using a thermometer or putting my hand on you. So that is a sign. If you have a rash on your body, that is also a sign. If you have a sore throat, that is also a sign. Where does symptom come in? With symptom, only the affected person can tell you about the symptoms. An example of symptom would have been that you are feeling dizzy. There is no way I can look at you and know that you are dizzy. I didn't say that you are drunk. I said that you are dizzy. So if you are dizzy, you would have to communicate that to me. If your belly is hurting you or your tummy aches, now that is also a symptom. There is no way I could look at you or detect that you're having a tummy ache. If you have a headache, pretty much that is a symptom. So I love to say in my definition that a sign is something that an external individual or another individual can see or detect, while a symptom must be communicated by the person who is affected. Yes, fever is a sign. Stomach pain, that is going to be a symptom. All right, feel free to do the research, and always remember, Mr. Wilson is open. You can come back to me with anything. If I am wrong, I am always willing to correct it, providing that I have facts. All right, great. So let's move on here. We are looking at signs and symptoms for both diseases. We are looking at, of course, looking at hypertension and yellow fever. We want to look at the sign and symptom for both. What do you have there? Share it with me in the chat. Sign and symptom for hypertension and yellow fever. I do appreciate the comments that I got here because it would have given me an opportunity to clear up misconception. Those persons who are on the edge, they are now better positioned, and those persons who are better positioned, they are even better positioned. So let me see what is happening here. Yellow fever sign, bleeding from the nose. Symptom will be headache. Symptom of hypertension, fatigue. Yes. All right. So Philip putting something out there. Philip warming up. I remember this morning at 5 o'clock when Philip joined, he thought that he was going to fail. But look what is happening now. All right. So here we go again. So sign is the physical look, the change in color, etc. Symptom is the body response to a disease. For example, coughing. This is sort of a little technical. Coughing. I don't know why you cough. I can't speak to coughing. That seems to be bigger than my pay grade. I'm sorry. Yellow fever, high temperature, yes. High temperature, hypertension, shorter breath. So there's a lot of things surrounding these two diseases. One of the things is that the one that I was looking for yellow fever is that you are going to have black vomit. So you are going to vomit, and when you vomit, it is going to be black in color. Now, the next thing I was looking for is that you are going to be having albumin in the urine. Now, remember that yellow fever affects the liver and the kidney. Sadly, I don't know that there's any treatment for yellow fever. No cure for it. I don't know that there, but you can get medication for the fever, but I don't know if there's a cure or it just has to do its time. As it were, for hypertension, signs and symptoms would have included headache, and you could have shortness of breath, and of course, there is a possibility that you could have nose bleeding. All right, so nose bleeding is associated with hypertension and not yellow fever. I've never seen that for yellow fever, but we have black vomiting for yellow fever. Can you imagine you? And then when you check it out, it is all, you're going to have headache as well, and you will be sensitive to light. Yes, so the list go on and on. Yes, those are also true. All right. So I was playing this morning as precursor to the class, my video on immunity, and we are supposed to look at this. I don't know if you are going to manage this, but I'm going to throw it at you. A lot of students are not enjoying immunity, and once I do the video, I would have picked up that students are afraid of something. Now, explain how vaccination works to prevent a person from becoming ill. Now, those persons who are doing both biology and HSB, you would have realized here that this is both a biology and HSB question. All right, so explain how vaccination works to prevent a person from becoming ill with yellow fever. So you want to take a vaccine so that you don't suffer from yellow fever, but we want to understand how the vaccine works. Tell us that in the chat as we come down to the end of the lesson. We are almost there. We have one more question to go, of which I'm going to be giving you the groundwork as to how to answer the question. It's too long for me to answer here, so I'll just give you the parameters to answer the question, And then we are going to hop on into number 6, which is sort of activity driven. We are going to talk a little bit of this, a little bit of that, and then we are going to do the final workout, that genetic diagram. You know I like genetics. I'm going to do that genetic diagram for you. I am going to ask you to do it. Tell me the answer, after which I will do it. Oh, vaccine. How the vaccine itself work, or how a vaccine work? I want to say this, whichever way you want to answer it. I would learn from whatever way you answer it. So I'd love for you to answer it whichever way. All right. Question, sir. Would it be best to give a sign or symptom that is unique to the disease, or does it does matter? All right, when you are giving sign and symptom for a disease, let us look at the word species that we were exploring last night. The sign and symptom is always there, but suppose you go to the exam and you can't remember the specific sign and symptom for that disease. However, you know that fever is a sign and symptom of the disease, though fever is a sign. Sorry, let me not use those words loosely. You go to the disease, go to the exam, and you know that black vomiting is a specific sign of yellow fever. But you don't remember black vomiting. You remember that the person will have fever. You're not going to leave that exam and say, I don't remember the specific sign. Fever is also a sign. While fever might be a sign for many other diseases, you want to ensure that you put it in. But yes, it is always best to put in the sign that is specific to that disease. But if you can't remember that and there's a general sign for whatever disease, make sure you write it on the line. You can't be wrong. All right. The injection, all right, they inject the person, and remember that it's not all vaccines that are given by injection. I think polio is a drop. They drop the thing in your mouth with an injection disease in order to stimulate immune response. I sort of like that, but it is not spot on. But I see where you're going, and I suggest to you that you go and watch my immunity video where I cover all the immunity and I break them down like real fine for you to understand it. Vaccine can be, all of these answers that you're giving me is the very reason why I made that video. You're just brushing over the edge because you're afraid of it. Nobody is studying anything about vaccine, but after you've watched my video, you are going to be fine. I'm going to answer the question for you. Vaccine is just let me put this on the screen. See how we can treat with it. All right, so vaccination is the artificial active way of introducing antigen into the body so the body can produce antibodies, some nice words coming up here, against the disease. This is usually done orally or injection. Philip, you come out nice and strong. I don't know why you're going in so really wicked pan the rest of persons in the room. You start coming out a kind of way really strong on them. I think I like it, Philip. I think I like it. It looked like some work went into that. All right, vaccination gives them small content of the disease, that is true, so that their WBC, I'm not too sure if I know what that is, will fight, their white blood cell, forgive me, I am so much of an old man, that their white blood cell will fight those and create a specific antibody. I see a lot of persons saying a lot of things and they sound good, but there's something that is very important for this particular question that I am not seeing. In the process of vaccination, artificial, all right, I am seeing that. All right, so let me sit down. So individual, but allow the lymphocyte to produce antibodies to fight. All right, I am seeing that. That not so bad, but I am still looking for a little something. All right, let me look at what is coming on the screen now. Vaccination give a person in all right, so we're getting some little stuff around the corner that we see on Google somewhere. When the teacher was teaching about vaccine, where the teacher never teach about vaccine, the teacher tell you to go and read on vaccine because the teacher just couldn't bother teach that the topic there, and the teacher feels that you could have done that on your own. So let me see if I can add to what you have there. A vaccine is a weakened form of the pathogen, right? And the pathogen, how it's weakened is that they could use formalin to weaken the pathogen. This is not a part of the answer that I'm telling you now. I just wanted to understand what a vaccine is. So they could inject it a couple of times and re-inject it and re-inject it to make it become weak. Or they could apply heat to it to cause it to become weak, or they could use formalin. All right, great. So vaccine pretty much, this is the answer now. Vaccine is a weakened form of the disease which provide artificial active immunity. That is what it does. Well, how does it do this? Now, the vaccine is of course it of course introduce pathogen to the body, and the pathogen will carry with it antigen, as a lot of person would have said. Now this antigen will of course cause the body to, or the white blood cells, to create antibodies that are specific to that type of a pathogen. But this is the part that I am not seeing. So you say all of that and it was said nice. This is the part that I am not seeing. And let me tell you why this part is important. Just hold one bit. I'm going to tell you. Look at this. Explain how vaccination works to prevent a person from becoming ill with yellow fever. So we are talking about becoming ill, or the next time you are exposed to it, why you won't become ill. This is why you won't become ill. Because the lymphocyte will make what we call memory cells, so that if you become exposed to this pathogen, what will happen is that these memory cells will understand, because they have that antibody, they know the antigen, they have the antibody, and they'll fight it just to prevent you from becoming ill with the disease. Very good. I think that the attempt were really, really wonderful. And I hope that I strengthened here. All that I added to what you said was that it creates memory. It has memory now as to what the antigens are, and as such, it has antibody to immediately fight it. So rather than running down the road to get some drugs at the pharmacy and there's traffic on the road, the body can already deal with it because it remember and it have a stockpile of antibody waiting for anything like this to come. I can produce it quickly and get rid of it. All right, that is just a big deal there. All right, discuss 3 strategies that can be used by hypertensive patients to control their signs and symptoms of hypertension. Now, if you observe here, this is not asking for also memory cells. Okay, great. This is not something that we are asking the doctor. We're asking what you can tell your grandmother to do, what you can do to control hypertension. Now, the first thing you are going to do is to eat healthy. That's the first thing you are going to do. You are going to have a lower salt intake, so decrease the salt intake. You need to maintain a healthy weight, so you will have to watch your BMI. Another thing that you will have to do, you'll have to exercise. Great, I've seen some nice thing coming up. Another thing that you will have to do, avoid drugs, which include alcohol, and you really don't want to smoke. So I want you now to expound on all those points that I've just given. It's value 6 mark and it takes up a whole lot of the paper. So I decided that I would have just given you some pointers here as to how to answer this. I gave you more than 3, and then you can expound how salt cause hypertension and how you deal with that, how the fat will cause it, how the sedentary lifestyle, and you do the discussion. I hope that you would have watched my earlier videos to understand what discussion means, what explain mean, and all those words that you are going to find on the exam paper. But you must know what describe mean, and when I meet you probably the next time, I'm definitely going to go in depth with this. Because if you observe, most of the questions here are asking you to describe. So you don't want to go to the exam and not know how to describe. So here again, let's describe. Describe how the human skin help the body to keep cool on a hot, sunny day. All right, tell me about that in the chat. How the skin help the human body to keep cool on a hot sunny day? Tell me in the chat. This is a wonderful question. I think this is pretty easy. I could make it easy. So we are looking at the concept of vasodilation and vasoconstriction. Choose the one that you prefer to answer this particular question and answer. First, you have to tell me which of the category your answer is under. Is it under vasoconstriction? Is it under vasodilation? Which of it? Which of these you're answering from? So let me just look at that. Here becomes erect. So let us look at the question again to see if we are going in the right direction. We're asking you to describe how the skin helps to keep the body cool. So we want to keep the body cool on a hot sunny day. So if we want to keep the body cool on a hot sunny day, we would have to revisit this answer because it's not pretty much in keeping with what you'd want the answer to be. Sir, we have to know about this for exam, like skin. I don't even remember what is on the broad topics. If somebody wants to tell me that, I'd be very grateful. I really don't remember that bit. There are some of the stuff I remember right off the bat. There are some I don't remember. So I am not going to say yes or no to that. But what I know is that these things are usually on could be featured on your paper one. All right. So what we're going to have happening on a hot day, we are going to have vasodilation. Now, vasodilation is dilate, mean that the blood vessels become larger, the blood is flowing closer to the skin, the body is sweating more, and all of those. Release of sweat, yes, sweating, the blood vessels, yes, the body become red, metabolic rate decrease, yes, it is not on the broad topic. All right, so Peter Gay said that it is not on the broad topic. So when I see that, I'm just going to move on. Guys, be careful of this broad topic thing, I'm going to show you the other question. And if I show you the other question and you tell me that you couldn't see this, this is not going to be on your exam or can't be on your exam, then you'd be fooling yourself. I heard Curran talking about this, the trick with the broad topic thing. Now look at this question. State one similarity and one difference between human being and invertebrates in the way they cool their bodies on a hot day. All right, being that you say it's not on the syllabus, I'm going to answer it very quickly. So we regulate our body temperature internally, while an invertebrate would regulate its body temperature externally. So if they are too cold, they'll move towards warmer area. If they are too hot, they are going to move to shaded, cooler area. And that is just a summary of what it should be. Let me move on down now to the big question that completes the paper for this morning. All right, so you are seeing the question on screen. Could you read and work it for me? So I am not going to be working it yet. I'm going to be looking at your answers. And when I look at your answer, depending on what is coming up, I should get a pen and work it on my paper that I can quickly say yes or no. Okay, I got a pen. Now you are going to work this and tell me what the answer is. And I'm going to be prodding you right through. I want to find out what you did because I think last night we did a question. I think the night before we did a question on genetics. So everybody should be mastering genetics now. All right, great. Okay, so girl, are these the topics? All right, nutrition, abiotic, biotic food chain, predator-prey relationship, decomposers, treatment and control of disease. Okay, excretion. I beg your pardon? Hold up, hold up, hold up, hold up, hold up. So the topic we were covering about the skin is excretion. Yes, eat, you're getting rid of eat, that is excretion. You're getting rid of salts and sweat, that is excretion. And that is why I'm saying to you, be careful of these broad topics that we're talking about. Try to understand the span of a particular topic. For example, excretion. Excretion will cover the skin. So be careful, guys. Yes, I want to go with the skin to say that the skin could be on it. Just let me look at this one more time. Nutrition, abiotic factors, yes, predator-prey relationship. And predator-prey relationship could take in the other type of symbiotic relationship as well. Decomposer taking the carbon cycle. It's a big exam, treatment and control of disease. How the disease a while ago. And transport excretion. So transport include circulation in both plants and animal. Xylem, and you could talk about the storage and all of that in plant. Yes, all that we have been doing so far. Excretion, reproduction, continuity, and variation, which is what we're going to do now. And continuity and variation include evolution. Okay, I'm seeing Peter Gay Campbell out of the block. I'm seeing a lot of things happening here, you know. Holy moly, holy moly, holy moly. All right, should I solve now? Should I solve with you now? Is everybody finished working this question? I don't know the answer, so I can't tell you what it is. I'll have to go work it with you. What? Okay. Okay. You see those persons who are saying yes, sir? You can't do the question. And I would suggest that you go over to my channel and watch it. All right, watch the genetics playlist. All right, before I answer the question, I want to share some things with you guys. Your genetic question could take any of the following tone. You could look at inheritance of sex. You could look at inheritance of dominance. So when we talk about inheritance of sex, we are talking about the next time a woman who has 1,000 boys, if she get pregnant another time, will she have a girl or a boy? And this is always going to be a 50-50% possibility. We could look at inheritance of dominance. It's the final question we're on the paper, but I have to give you a lesson or a crash course on the genetic topics that might appear on your paper. All right, so just allow me to do this before we make our leave. And I have a whole lot of stuff to do here after. I don't know if we're going to see each other later. If I can, I will. If I can't, just forgive me. I'll try to put some videos out or something. Or you go on over to the channel and watch something. I don't know if it is wrong, but let me just go with the many stuff. So I spoke about inheritance of sex. That's the first one. Then the other one is inheritance of dominance. In order to do the inheritance of dominance, all of these are the same crossing, but it's just the laws that go with them. With inheritance of dominance, what the law says is once the allele for the dominant trait is expressed, then the phenotype will be that of the dominant trait, right? And I think I explained this. I think I have about 3-4 different videos on the live videos that I've done on genetics. Then we have inheritance of recessiveness. In order for you to acquire a recessive trait, you must get in its homozygous recessive form. That is 2 small letters. Then we have sex-linked inheritance, where this is the one that you have to talk about both the disease and the sex of the individual. With sex-linked inheritance, there are 2 things on the syllabus that will appear or more than likely appear on the exam. It's either they are looking at hemophilia, which is bleeding disease, or they are looking at color blindness that we did last night. For recessiveness, they are looking at sickle cell anemia or albinoism. Now, not only should you know the cross for sickle cell anemia and albinoism, all of these that I made mention of just now, hemophilia, color blindness, you need to know what they are. You need to know what is sickle cell. You need to know what is albinoism and what are some of the precautions that are to be taken. Let's move on. There's also codominance and incomplete dominance. With codominance, as it were with the ABO blood group, or as it is with the impatient, with codominance, the trait, the phenotype expresses for both trait. So if it's codominant and you mix black and white, the offspring will come with black and white. That's for codominance. For incomplete dominance, if you mix black and white, you will get gray. If you mix red and white, you will get pink. Then there is also what is called crossback or test cross. I haven't seen that on the paper, but it's one of those things on the syllabus. Then there is what we refer to as the pedigree chart. I have never seen a CXC book that covers it properly. So I went out and I compiled something on the pedigree chart. If you go to my channel, it might better assist you in solving a problem with the pedigree chart. It is on my channel. Go and watch a video on pedigree chart. So let us look at this particular question to be answered. I haven't even read it as yet. So I'm going to just small up my screen. And as I go through this, I want you to take out your pens and pencil. And I want you to write with me as we go through. We are going to solve this question exactly like you are supposed to solve it for CXC. Here we go. Persons with the inherited condition albinoism do not produce the pigment melanin that gives the skin its dark color. Use the symbol A to represent normal allele. So what I'm going to do, I'm going to start by defining my allele. So I'm going to say remember, I'll just spell it out and say it and you write it. So I'm going to say let this capital A be the normal allele. You write it out, I might not be able to. Let it be the normal allele for albinoism. While we are going to use common A, and common A here represent albino. So this is pretty much the recessive allele. That's a recessive allele. So the first thing we need to do is to define the alleles. So that is step one. Let us continue. Draw a genetic diagram to illustrate how two heterozygous parents could produce an albino child. So the first thing we know here, we have to now write the parental phenotype. So you are going to write the parental phenotype, and the parental phenotype is what the parents are. So we say that they are heterozygous for the trait, and I don't know if they use carrier here, but they are heterozygous for the trait. I think they are carriers. They are carrier for the trait. All right, so these are two carrier normal individuals. They are heterozygous for the trait. So you would write that out here. That mommy is a carrier, papi is a carrier, or there are two individuals that are heterozygous for the trait. Having done that, the next thing we are going to write is the genotype. Make sure that you are writing it on the paper in this order. So this is 2 and this is 3. Genotype. Now we know that the genotype, based on what is given here, it is heterozygous, which means that capital A, one parent is going to be capital A, common A, and the other parent is going to be capital A, common A. All right? In this case, it's frankly, you really don't need to name whether it is a male or a female. That is applicable when we are doing sex-linked inheritance or inheritance of sex. All right? So because we're not going to know whether it is a boy or a girl, the offspring is here. So don't confuse yourself about boy and girl here. That is not important. All right? This is inheritance of recessiveness. After we have done this, then we go to step 4. Step 4 is where we are going to write out the gametes. All right. All that we do is to separate, right? These are a little further apart. That's going to be the gamete. So all that we do is to separate it here. Great. So that is step 4, right? The gamete. So step 1, you define the allele. Step 2, you write out the parental phenotype. Step 3, you write out the genotype for the parent. Step 4, you write out the gametes. Step 5, this is where you are going to now have segregation or random fertilization. So this is the area now we are going to do the cross with the lines, or you are going to do the Punnett square. Everybody in the world who knows Mr. Wilson know that Mr. Wilson is going to do the Punnett square any day of the week. Because as you get deeper into biology, those line crosses cannot help you when you are doing dihybrid and trihybrid. What we are doing now at CXC is monohybrid inheritance. So we are pretty much looking at one trait, monohybrid inheritance. When you go up, you are going to be looking at 2 traits, dihybrid inheritance, and then you are going to be looking at probably 3 traits, which is of course trihybrid inheritance. I think I'm not as good as I used to be because I can't remember the symbol for male and female. So I'm going to try, and you can tell me no if it is no in the bracket. How is right again? I think the male is I think the male, help me here. The male is a circle with a cross. If you don't remember, just write out the word male. And the female is a circle, I think, with a triangle. That might be it. I don't remember. Remind me if I'm doing nonsense on the screen. But does it really matter? No, it does not. Because here, male or female doesn't matter. All right? So we're just going to put one of this here. That's an A. And we're moving this one up. So we do that there. It doesn't really matter if it's male or female because we're not looking at the sex. And then we are going to put this here. I'm not so efficient. And then we'll put that here. Now remember wherever they intercept. And this is step 5. We are doing step 5. That is actually step 5. So wherever they intercept, we are going to write everything. So here we go. Those 2, they intercept here. So we know that this is going to be a normal individual. These 2, they are going to, this is heterozygous for the trait, and we could say that they are carrier just like the parents. And then again, this is going to be heterozygous for the trait. Very well. And then these 2 intercept here. So we have common A, common A. All right, great. So let us see what the question is saying. Oh, first, I need to change the color and number the parts. So this here is going to be 1. We're going to use that as 2. And I'm just labeling it that I can question you and make your life a little more difficult. All right. So now I'm going to go back to the question somewhere up the top there. And the question, what it wanted us to do, it said that using that, we are supposed to illustrate how 2 heterozygous parents could produce an albino child. So here, the albino individual, tell me in the chat, what number represents the albino individual? Oh, it's buffering, I'm sorry. Oh, you students gaze when the teacher is working and talk about other stuff. You have to use your time wisely. You can't do that. You need to pay attention. I've been seeing that since yesterday. Once I am not here and I'm on the board writing, persons are talking about everything else. All right, great. So 4 there is going to be the albino child. All right, let me see if there is anything that we should have answered there that I did not answer. So 4 is going to be the albino child. And it says using this symbol A to represent normal allele and common A to represent the albino, draw a genetic diagram to illustrate how heterozygous parents could produce an albino child. All right, so there, so we could just identify, writing out that this is the albino child. Then it said, identify the genotype for the albino child. The albino child, could you write the genotype for the albino child in the chat for me? What is the genotype for the albino child? Genotype for the albino child. Write it in the chat there for me. Okay, great. A lot of persons coming in nicely. All right, so the albino child, the genotype is always those letters. So AA is going to be the genotype for that particular child. All right, great. So the albino child is this particular child, AA. All right. Now, let us look at what the other question says. It says that we're supposed to state the probability of the child, state the probability of the albino child being produced. Now, what is the probability there? State the probability of the albino child. What's the probability? So there's a 25% probability. I'm seeing that coming out. Hold on. No, it is not a 3-to-1 probability. It's not a 3-to-1 probability, you know. It would have been more a 1-to-3 probability than a 3-to-1. So it's a 25%, but not a 3-to-1. All right, so it's not 3-to-1 either. It is going to be 1-to-4. So right here, no. The ratio is going to be a quarter. Not the fraction is going to be a quarter or 25%. And I'm seeing persons writing. Let us answer the question that is asked and not all the things that we learned some time ago. The probability of a child becoming albino here is a 25% chance that the child might become an albino. All right, all the other fancy stuff, learn to use them, just answer the question that is asked. The probability here, the easiest thing, 1 out of 4 is 25%. And I'll show you, 1 out of 4, 25%. If you put this over 100 over here, all right, 1 cancel 1, 4 into itself 1 time, 4 into this will go 25, which give us 25%. So it's a 25, the probability of the child becoming an albino, there's a 25% chance or a 1 in 4 chance, right? There it is, 1 in 4 chance or a 25% of a child being produced being an albino. That is 6 mark, easily achieved there. It takes us pretty much to the end of the exam or the end of this paper. It was nice being with you, but in the future, if we plan to have an early class, try as much as possible to be a part of the early class. I can't stay much longer with you because I have other things to do, and that is why I put you in here. But like I say, I'm really happy that you took the time to join the class. It says a lot to me that you want to learn. We're going to try our best to see. Well, I'm working alone now. I'm going to try to see how best I can pretty much put the content in video for those persons who need to revise. I understand that persons are learning at really different speed. So because you're learning at different speed, there are some persons who are doing well here, but there are some persons who are going to need a little more assistance. Now we have just 2 more papers for you. We have the 2018 paper, which I have not looked at as yet, and I have the 2021 paper. I don't know if you see me this evening, I will be doing the 2018 paper, and tomorrow it would be nice if I do the 2021 paper with you. You are going to have the first show, and we are going to try to see if we can have a show for the HSB students so that we can start their preparation, and your preparation would have just been at the end. Somebody sent me, I'm seeing some broad topic that you sent just now. There's something that I could have, there's something that I can do. I think I could probably go up until 9 o'clock. I could probably look at some of the content around the broad topics, some things that I have been seeing students having problem with. or I could share with you some videos to correct some of the ills that you have. If I share with you some video, I am going to be resting my throat and you are going to be learning. You still can talk in the chat. So what I'm going to do, I'm going to see if I can share the video with you before I do anything else. But I'll be here. I just want to rest my throat that I have it to help all the students. All right, so we started out with I don't want to do pedigree chart now. You could go and watch it, and it's not a long for what I want to do now. I want to just do something that is pow, spot on. What I said I wanted to show you just now. All right, great. We did type of immunity this morning, so I'm not going to do that now. All right, you have a video coming up now. You said that this will be on the exam, so there we
Symbiosis. Symbiosis refers to an interaction between organisms of different species. The interaction may bring benefit, harm, or no notable advantages nor disadvantages. Mutualism. Both organisms benefit from the interaction. The interaction between a ticks and a cattle would have been parasitic. However, the egret removing the ticks from the cattle would have formed a mutualistic relationship in which the egret would have benefited from getting food and the cattle comfort. The sea anemone and the clownfish, as is seen in the picture. Here, the clownfish would have gotten food and protection from the anemone, while the anemone would have gotten nutrient from the dropping of a fish. The fish would also remove organisms that would have, of course, harm the anemone, which makes it a mutualistic relationship as both organisms are benefiting. Then there is also the nitrogen fixing bacteria that is located in the root nodules of leguminous crop. Here, the bacteria would have gotten somewhere to live while it is fixing nitrogen that the plant is able to use for its growth and other processes. Shark and the remora fish. This is, of course, another mutualistic relationship. Here, the remora fish would have actually hitched a ride on the shark, moving around freely, no energy used, while it would have been eating the scraps left over from the fish, thus providing this clean environment and safe environment for the shark. This is a good example of mutualistic relationship. We're seeing the egret there on the horse, but the egret is popularly found among cattle, and the cattle are standing over there wondering, why is the egret over there and not over here? We have work to be done. We need some ticks to be removed. Predation. Predation is popularly called a predator-prey relationship, and it refers to a relationship in which one organism, the predator, hunts and feed on another organism, which is called the prey. So here the shark would have been the predator and man the prey. And there's also this relationship between plants and herbivore, where the herbivore is the predator and of course the plants would have been the prey. Here we have another relationship with the beetle here and the aphid. So the aphid in green, and we refer to this beetle here as a love bug. So this would have been a relationship in which the love bug would be eating the aphid here. A biological control in agriculture. The aphid would be, of course, be parasitic to the plant, as it would have been taking sap from the phloem vessel in the plant. Commensalism. Commensalism refers to a relationship in which one organism benefits and the other is not affected, as in the case of a hermit crab using the shell of a dead snail. The snail is not affected, but of course, the hermit crab would have gotten protection from the shell. Another example here is the egret, which follows behind a grazing cattle. Now, in this particular relationship between the cattle and the egret is not really feeding from the cattle. As the cattle moves through the grass, the egret would, of course, feed on whatever is found in the grass and not on the cattle. Here, the egret is benefiting, but the cattle is in no way affected. Then there is, of course, the epiphytes on trees. And epiphytes would be like this plant here growing on another tree. It is there getting sunlight, but it is not affecting the plant in any way, shape, or form. So it benefits from this high elevation. No predator would be affecting the red plant here seen on this tree. However, it would have been able to carry out photosynthesis without hurting the plant on which it is perched. This is a popular one, parasitism, and this pretty much refers to a relationship in which an organism is, of course, hurt while the other organism is benefiting. So here the mosquito is a parasite to human here having a grand time, as they are, of course, sucking the blood from the human and could very well introduce the plasmodium, or one would say could cause dengue or malaria. Now, the mosquito is harming man. Tapeworm and man is also another example of a parasitic relationship. And of course, the ticks on the cattle or on human is a good example of a parasitic relationship. Here we have a ticks, and this is really dangerous to both cattle and other organisms. Amensalism, this is not on the CXC syllabus, but it refers to a relationship in which one organism is hurt or destroyed while the other is unaffected by the interaction. So the elephant here moving through the grass is of course affected. The elephant is not affected in any way as it just moves pretty much through the grass. Penicillin pretty much killing a bacterium. Here, this is a case where the penicillin is not affected, as penicillin is pretty much from a fungus. So the fungus is not affected, but the bacteria is, of course, killed. And another thing we could look at is competition. Here we could have intraspecific and interspecific competition, where intra speaks to a competition within the species and inter with different species. And then we have antibiosis. Antibiosis speak to a relationship in which one organism secretes a chemical to kill another. The last one we look at here is neutralism, and this is not on the syllabus for CXC. Here, organisms, they are usually found together, but they benefit nor affect each other. They are just there, as we would find plants in the desert. This takes us to the end of the presentation for today. And of course, please be reminded to like, share, and of course, subscribe. Also be reminded that you can benefit from our many past papers on the channel.
introduction. Today we're looking at types of immunity, and of course I am Mr. Wilson from CSEC Biology, TCP. There are two types of immunity: active and passive. Let's look at the animation for active immunity. Active immunity occurs when an individual is exposed to a disease which causes the immune system to produce antibodies specific to that disease. It is important to note here that the individual would have been exposed to the disease, and that immunity is of course being developed as a result of action within that person or organism. So here we have a fellow being introduced to the ranch, and with him he brought pathogen, and you're seeing the pathogen populating the screen in red. And as the pathogen enters the environment, you're seeing that the lady is developing some antibodies that are of course going to be guarding her against this particular pathogen. So of course she will not become ill as a result of her building her defense from within. So here she's exposed to the disease, and as such she would have developed some resistance. With passive immunity, what happens is that the antibodies are of course given to the individual rather than them producing it through their own immune system. So in the passive, you're seeing that it is given to the person, while in the active, it is the person who is actually making the antibody. So in passive, the antibody is made elsewhere and given to the individual. However, in active, it is of course made within the individual. And we're going to continue to reinforce that throughout this video. Here we have examples of passive vaccine. There we're seeing, and of course a mother with the fetus, and we're seeing a mother breastfeeding. Now, immunity could also be considered as natural or artificial. With natural immunity, it is organic. Natural innate active immunity. This is where we start now to think about our exams. Now, these words are all words that they carry specific meaning, and of course direct you to the particular immunity. So natural, it is organic. It has no influence of man. Now, this type of immunity is an immunity that you are born with. How do we know you were born with this immunity? It is innate. That suggests that you were born with the type of immunity. Now, where do we get the antibodies from? Active here tells us that you are making the antibodies. So whenever we see active, it means that the antibodies to safeguard you against that particular pathogen is made within the individual and not with external assistance. Then there's also natural innate passive immunity. And let's decode. Natural, it's organic, it has no human interference. Innate, meaning that it is made within a organism. And of course, passive here mean that it is given. It's something that is given. It is passed from one person to another. When passive, we talk about passive, you are given the antibodies rather than you making the antibody. So here, the offspring gets the immunity from the antibodies that are, of course, produced by mom that crosses the placenta to the fetus. So the antibodies are not made in the baby. The antibodies are actually made in the mother, and of course passes the placenta to the baby. The baby also acquires some amount of immunity from the breast milk. So passive here suggests that you were given the antibodies. It's not that it was made within you. So the baby was given the antibody. It's not a case that the baby is able to make the antibodies for itself. Now innate, of course, when the baby is born, or it is made within. Innate, when the baby is born, will born that with a short-lived immunity, and it is, of course, innate speaks to being within the individual. Why is this youngster running? It seems so happy. Of course, he would have acquired acquired active immunity. What does that mean? Acquired, the immunity that develops after birth. So immunity, if we were supposed to compare, we would compare acquired to innate. So if it's from birth, then it's innate. If it's acquired, then it's after birth. If we were supposed to compare natural, it would be compared with artificial. If we were supposed to compare active, it would have been compared with passive. So this type of immunity is one that develops as a result of exposure to the disease. Example, smallpox and tuberculosis are examples. Now, this youngster probably would have been exposed to the disease, and having exposed to the disease over time, he would have gotten some immunity. So there's a great possibility here that he is now told that he's no longer suffering from the disease, he's immune to the disease, because his body would now be making the antibodies to fight off the particular disease. Who wouldn't be happy? Hee-hoo! That's what most persons would be saying. Now, artificial immunity is one that is, of course, introduced, or it has direct human influence. So whenever we go for vaccine, that is a good example of artificial. Now, it could be artificial acquired active immunity. And let's break this down. Artificial meaning that it has some human influence. Acquired meaning that it was, of course, developed after birth. So you were not born with it if it is, of course, acquired. And then active, even though it was given to you by a human, it is within your body that the antibodies are going to be made. So with this type of immunity, artificial acquired active immunity, immunity that develop as a result of individual being given a weakened form of the disease. And the disease here is weakened by eating, or we could use formalin, or we could re-inject several times, and of course weaken the pathogen to a manageable form that can be injected in someone. Usually it is administered through vaccine. Examples would have been that for tuberculosis, polio, and BCG that we would have received in the early years of our life. It's very important for you to pay attention and look at the photographs that we're using in the pictures, as they are giving you prime examples of each of the type of immunity. Now, artificial passive acquired immunity. Artificial here, human influence. Passive meaning that the person was exposed to the antibodies and not to the disease. So here the person would have been given the antibodies because it's passive, and acquired meaning that this resistance, this immunity actually developed after birth. So we have both on the screen, and we continue to emphasize that when you get to the exam, you can break down this long name, artificial passive acquired immunity. What does it mean? And if you were supposed to see passive acquired immunity, what does it mean? And if you were supposed to see passive immunity, what should we understand from this? So here we have broken it all down. We have given it a meaning for all the words in the names. So you should be able to map this nicely when you get to the exam. Now you're seeing this, and this one is synonymous with bites from snake. Now it is acquired through the introduction of antibodies directly into the individual affected by the disease. And usually you would have seen my first video on serum and plasma. Here we have what is called antiserum, which is used to treat this type of problem. It is fast acting but lasts for a short time, and it is used during cases of emergency where somebody is bitten by a snake or where the person is suffering from tetanus. This type of a vaccine is administered so that there is quick response and the person could of course recover quickly. If you look at this young man, he was bitten by a snake probably, and of course he's been injected probably with of course antiserum to ensure that he will survive. So looking at the snake, it reminds us that it is artificial passive acquired immunity. We want to ensure that we are paying attention and be reminded that you could go over the slide however many times you so desire, so as to make sure that you understand well that which we have covered. We have just looked at what's the difference, and we're looking at type of immunity, one might say types of immunity. I am Mr. Wilson from CSEC Biology TCP for TCP Academy. Please be reminded to like, share, and of course subscribe. And when you do, click on that notification bell and select all. So
even though it is just the removal of metabolic waste from the body, which would otherwise poison the body. In this lesson, we'll be looking at the nephron, which are found in the kidney. The kidney is one of the body's major excretory organs and is responsible for cleaning the blood and maintaining water balance as it produces urine. However, the kidney would be of no use without working nephrons. This lesson is suitable for all students studying the topic excretion. I am Mr. Wilson from CSEC Biology, the cover page. Let's explore the nephron. The nephron lies between the cortex and the medulla of the kidney. The nephron happens to be the kidney's functional unit and is also called the kidney tubule. Being the functional unit, it is pretty much the site of operation. If you were supposed to think of a motor car, in order for that motor car to drive, the functional unit would of course be the engine, which would of course provide that energy for the car to move from place to place. If you were supposed to think about a country, that mega city, if you were supposed to think about that major town that drives commerce, for Jamaica it would have been Kingston. So it would have been the functional unit for Jamaica. The nephrons carry out ultrafiltration and selective reabsorption during the production of urine. Hence, the blood is cleaned and the water balance of the body is of course maintained by the nephron. The nephron, like I said earlier, lies between the cortex and the medulla of the kidney, and of course it empties urine in the pelvis. Now here we have a diagram of the kidney, and we're looking at the kidney from the back of the kidney here. So you'd observe at the bottom of the screen you're seeing the red line here representing that renal artery, the blue line here representing the renal vein, and of course you'd have seen the pelvis, which is just an enlarged portion of the ureter. The nephrons have Bowman capsule, proximal convoluted tubule, and distal convoluted tubule in the cortex. The cortex has a lighter color than the medulla and is closer to the part of the kidney which is of course distal to the attachment. The medulla is closer to the attachment. So we are saying here that the cortex here is located closer to this portion of the kidney. But can you identify the Bowman's capsule, the proximal convoluted tubule, the distal convoluted tubule? How well did you do? Let's check it. Bowman's capsule, first convoluted tubule or the proximal convoluted tubule, second convoluted tubule or the distal convoluted tubule. Now, proximal convoluted tubules speak to the tubule being closest to the Bowman's capsule. Proximal convoluted tubule closer to the Bowman capsule. Distal, it's a distant away, some distant away from the Bowman capsule, which makes this the proximal convoluted tubule within proximity and distal some distant away, or first and second convoluted tubule. The nephron have loop of Henle and collecting duct in the medulla. The medulla is darker in color than, of course, is the cortex, and it is closer to the attachment on the kidney. So here the attachments are. At the bottom you have the pelvis here, and you have the blood vessels here. However, the loop of Henle and the collecting duct is of course located in the medulla. Can you identify the loop of Henle? What about the collecting duct? How well did you do? Let's check it. Loop of Henle, collecting duct. It's very important for you to know the parts of the nephron, as the parts of the nephron carry out different functions. Let's explore the parts. The diagram shows the nephron with the network of blood vessels that assist the nephron with its operation. The nephron or kidney tubules have a network of blood vessels which are wrapped around the tubule. The blood vessels along with the nephron facilitates ultrafiltration, selective reabsorption, and of course maintenance of water balance in the body. The Bowman's capsule becomes a major part of the nephron as molecules from the blood vessels in the glomerulus are forced into the Bowman capsule by ultrafiltration. These are some of the molecules that are forced across the membrane into the Bowman's capsule: amino acid, urea, glucose, salts, water, and of course hormones. Now let's look at what happens in order to get these molecules from the glomerulus into the Bowman's capsule. And you'll observe on screen that these areas are labeled. Now the arteriole entering the glomerulus is larger than the arteriole leaving. Now if you observe your screen, you'll see a pulsing circle showing the blood vessel labeled the afferent arteriole. If you were supposed to observe closely, you'll observe that this blood vessel is of course larger than the blood vessel that is leaving. Now the blood vessel that is leaving is called the efferent arteriole. Now because of this difference in the size of the blood vessels, it creates a pressure in the glomerulus that squeeze substances through the capillary into the Bowman's capsule. The process is known as ultrafiltration and is the first step in urine production. There we have the glomerulus, that network or knot of blood vessel through which the substances are squeezed into the Bowman capsule. So we're seeing the afferent arteriole being the larger arteriole here, and the efferent being the smaller arteriole, creating that pressure within the knot of blood vessel called the glomerulus, causing substances to be squeezed through the membrane into the Bowman capsule. But what happens in the proximal convoluted tubule? In the proximal convoluted tubule, which is highlighted on screen, or the first convoluted tubule, selective reabsorption is evident as useful substances are reabsorbed from the filtrate in the tubule back into the blood. Now, glucose, hormones, salt, water, amino acid, and of course, vitamins are all reabsorbed in the first proximal convoluted tubule. It's important to note that some substances that were not forced out into the Bowman capsule are forced out here. So understand that the tubule is that orange color diagram on your screen, and there is movement of substances between that orange diagram called the nephron and the blood vessels. And of course, it is seeping through the membrane from one side to another, from the blood vessels to the nephron, and of course from the nephrons to, of course, the blood vessels. It is very important to know that there are several nephrons in the kidney, which are of course connected to collecting duct. The substances continue to flow through the nephron from the Bowman capsule to the proximal convoluted tubule, and the selective reabsorption started. The substances are going to move into the loop of Henle. Now, selective reabsorption of water takes place in the loop of Henle. Now, having been reabsorbed due to a regulatory process and selective here, which suggests that it is just the amount of water that is needed that will be reabsorbed. If it is not needed, then it will not be reabsorbed into the blood vessel. The substances will continue to move into the distal convoluted tubule, or as we call it, the second convoluted tubule. Now, selective reabsorption of salts and water takes place in the distal convoluted tubule. And of course, this is regulated by ADH, which is of course known as the antidiuretic hormone. The substances will continue, having been reabsorbed, filtered, and ready to leave the body. And if you look at your screen now, you're realizing that there's an animation showing the passage of substances through the nephron, and of course passage of substances through the blood vessel to include blood. There's also a flow of urine down collecting duct. Now, urea, salts, and excess water are collected in the collecting duct as urine and moved to the pelvis to be excreted from the body. Some reabsorption of water, if necessary, takes place in the collecting duct, which is of course driven by ADH. ADH will affect the permeability of the collecting duct and of course the distal convoluted tubule, making sure that water balance in the body is maintained. There we have it. How much did you learn from this lesson? Leave 2 questions from the video in the comment. Other viewers will answer these questions which are based on what you learned. However, you are required to answer these questions just to test how much you've retained from the lesson. What is the function of a kidney? What is the nephron? What is the function of a nephron? Name the parts of the nephron. Distinguish between ultrafiltration and selective reabsorption. Distinguish between the afferent and efferent arteriole. For 4 above, name one substance that is reabsorbed in each named part. What is the role of ADH in the nephron? Number 1, the kidneys, which is of course 2, clean the blood and maintain water balance in the body. 2, the nephron is the kidney's functional unit, which means that it is the site of operation. 3, the nephron's function is to carry out ultrafiltration, selective reabsorption, assist with water balance in the body, and of course make urine. 4, the parts of the nephron are Bowman capsule, first convoluted tubule or proximal convoluted tubule, loop of Henle, second convoluted tubule or distal convoluted tubule, and of course collecting duct. And this shows pretty much the flow direction of filtrate within the nephron. To distinguish between ultrafiltration and selective reabsorption. Ultrafiltration is a process in which substances are forced out of the glomerulus into the Bowman capsule from the blood, while selective reabsorption is a regulatory process in which substances are taken back from the nephron and allowed to re-enter the blood. The process could also see more substances be taken from the blood back into the nephron. 6. So distinguish between the afferent and the efferent arteriole. The afferent arteriole is larger than the efferent arteriole, and it takes blood to the glomerulus, while the smaller blood vessel, the efferent arteriole, takes blood away from the glomerulus. Now you're going to revisit the video to find out the substances that are reabsorbed in 4. Be reminded that ADH affects the permeability of the membranes within the collecting duct and distal convoluted tubule, causing more water to be reabsorbed when there is added ADH and less water to be reabsorbed when, of course, there is less ADH. Thanks much for watching. This is CSEC Biology, the cover page, and you've been listening to Mr. Wilson. Please be reminded to like, share, and of course, subscribe. Check out our 3 live classes weekly on Wednesdays, Fridays, and Sunday at 5:05 p.m. and we're streaming out of Jamaica in the Caribbean.